All-powerful God, you are present in the whole universe and in the smallest of your creatures. You embrace with your tenderness all that exists. Pour out upon us the power of your love, that we may protect life and beauty. Fill us with peace, that we may live as brothers and sisters, harming no one. O God of the poor, help us to rescue the abandoned and forgotten of this earth, so precious in your eyes. Bring healing to our lives, that we may protect the world and not prey on it. That we may sow beauty, not pollution and destruction. Touch the hearts of those who look only for gain at the expense of the poor in the earth. Teach us to discover the worth of each thing, to be filled with awe and contemplation, to recognize that we are profoundly united with every creature as we journey towards your infinite light. We thank you for being with us each day. Encourage us, we pray, in our struggle for justice, love, and peace. Our Lasallian prayer. I will continue, O my God, to do all my actions for the love of you. Saint John Baptist de La Salle, pray for us. Live Jesus in our hearts forever. Amen.
Hi everyone and good morning. Welcome to day 2 of TAAS 3, the Metabolomics approach and its different applications. I am Chris Argamino of the DLS-UCIF and I will be your host again for today. So roll call po muna tayo. Can you type in the chat box or comment section your location at the moment so we'll know where you are right now? Check natin . So again, kindly type your location in the chat box so alam natin kung taga saan po yung ating mga viewers ngayon. Wow! So we have Baguio, Pasay, Japan, Batangas, Davao, Quezon City, Bicol, Batangas, Pampanga, Tarlac, Navotas, Cagayan de Oro, Nueva Ecija, Palawan, Bulacan. So let me check from the Facebook chat. So we have one respondent from our Facebook comment section, and so Albay. So again, if you have friends na hindi nakapasok dito sa Zoom natin, the stream is available via our Facebook page. So may live stream po tayo doon. So we have Marikina, Cabuyao, Laguna. Ayan. Good morning po sa lahat. Sa mga taga-Bismay, maayong buntag. Okay? So and thank you for waking up so early for this webinar at sana nakapagkape na kayo kasi may kinalaman sa kape yung webinar natin today. We hope that you learned a lot in this session. So before we officially begin the event, may I remind everyone of the general privacy notice again and the house rules for the entire webinar session today. So run through lang natin. First, screenshots and video recordings of the event will be done and may be used for print, online, and social media publications. This event may include media coverage and story releases online, in print, or on traditional broadcast channels. Information collected via registration or media capture may be kept for archival purposes in the Google Drive account of the DLS-UCIF team. Fourth, personal recording of the session shall not be done to respect the rights of participants in the session. The recording of the session shall not be reshared and or reposted online without prior notice from the organizer to prevent unauthorized disclosure and processing of the personal data. Sixth, discussions and conversations of a personal nature not related to the ongoing session shall be discouraged to prevent unintentional and or unauthorized disclosure of personal data. So if you have personal stuff going on, maybe you can use other messaging apps instead of our Zoom chat. Okay, next, house rules. So for our house rules, the first one is that this webinar is being recorded and you will be able to view it on demand later via the live section of our Facebook page. So actually, the webinar last week is already available in the live section of the Facebook page. So if you want to recap what was discussed, you can go ahead and go to our live section at any time. Number two, at any point in the webinar, feel free to type in your questions via the Q&A box for Zoom and via the chat box for Facebook live stream participants. The speakers will try to address as many questions as possible during the open forum later. If time does not permit, our team will get back to you with the answers via email. For the fourth one, e-certificates can be requested by participants. So to emphasize, who have both pre-registered, so you must have pre-registered, and you must also submit the webinar evaluation form whose link will be sent later in the chat boxes after each session. The link to the webinar evaluation form or WEF will be posted later. And you can submit that until 11:59 p.m. tonight. Okay? So hopefully those who have pre-registered and evaluated us last week have already received their certificate. So if not, you can email us so we can address your concern. Okay, so before we proceed to the webinar proper, a quick recap muna of what happened last week. So for last week, we had the first session of the third installation of TAAS. In that session, we had two speakers. So una muna si Dr. Karen Lacerna, our DOST PCHRD Balik Scientist Awardee, and a guest speaker, Dr. Maria Fe Kalingason, who focused on rice metabolomics. So from that session, we were introduced to different omic studies and their hierarchies, including a rather in-depth discussion of metabolomics and its applications in a couple of aspects. So first, Biochar for Urban Agriculture. This was from Dr. Karen Lacerna. Combat trauma injury. So imagine a metabolomics application relevant to military/defense. No? So hindi pa siya nangyayari, I guess, sa atin dito. And lastly, earthworm metabolism, which had ecology implications. So we were also introduced to data processing techniques like the principal component analysis, correlation analysis, network analysis, as well as some instrumentation techniques, which we can use for the studies mentioned. From that session also from our guest speaker, Dr. Kalingason, we were also reviewed in genetics and obtained a couple of new information about our beloved grain, rice, including characteristics such as length, shape, gel, temperature, amylose content, and aroma, including global preference. So we also learned the role of metabolomics in rice research in applications such as single and multi-platform approaches and multi-omics approaches. These are only a few of the learnings that we had and we've gathered from last week. So now we're on to the second session and we'll be focusing now on food metabolomics in addition to the continuation of the introduction to metabolomics from Dr. Lacerna. So in this session, we're lucky to have two speakers again, Dr. Lacerna, and our guest speaker this week will be Dr. Emmanuel Garcia, who is an assistant professor at the DLSU Chemistry Department as well as the director of the LaSalle Food and Water Institute. Okay? So thank you, sir, for gracing us with your presence despite your busy schedule. Before heading to the webinar proper, let us hear some welcome remarks. Actually, we'll skip that part since part 2 na to. And we'll head straight to the introduction of the CIF through a recap of the video last week.
To the De La Salle University Central Instrumentation Facility. The facility is located at the DLSU Laguna campus in Binyan, Laguna, and it is the premier instrumentation center of De La Salle University. It is under the Office of the Vice Chancellor for Research and Innovation, and it aims to elevate the quality of scientific research, not only in DLSU, but also in the entire country. So the team or the CIF is manned by the following team composed of faculty, equipment specialists, and dedicated laboratory analysts. Let me introduce to you the laboratory equipments that are available in our center that will help you in your research needs. First, we have the sample preparation laboratory equipped with the common laboratory instruments that will help you process your sample, the microcentrifuge, semi-microbalance, freeze dryers, sonicator bath, water purification system, ultrasonicator probes, among others. Then the NMR laboratory is equipped with the most advanced NMR instrument in the country. It's 600 megahertz NMR instrument. It has a very powerful superconducting magnet, and it has a two-channel amplifier system capable of auto-shimming, on deuterium digital lock system, and as an auto-sampler. The NMR machine is equipped with a liquid probe, which allows us to analyze sample in solution in both 3 mm and 5 mm NMR tubes, and it is also equipped with a solid probe, which allows us to analyze sample in solid form. We offer services like the routine NMR analysis, 2D NMR analysis, multi-nuclear NMR analysis, multi-pulse experiments. We also do variable temperature experiments in the range -100 to 150. We do traditional inorganic multi-nuclear analysis, and we can do solid NMR analysis as well as copolymer analysis. We also offer trainings for basic NMR spectroscopy for chemistry and non-chemistry majors. We also offer training for structure elucidation using NMR spectroscopy, as well as advanced techniques in NMR spectroscopy. The spectroscopy laboratory is equipped with instruments used for spectroscopy. So the Shimadzu UV-Vis spectrometer is equipped with a reflectance attachment, which allows us to analyze not only solid solution, but liquid solution, but also solid samples. We can analyze reflectance because it's equipped with integrating sphere attachment, allowing us to analyze sample in solid state. Small and large samples are possible. We also have the FTIR equipped with ATR attachments, which allows us to analyze samples directly into the instrument. We also have the Malvern synthesizer, which allows us to analyze the particle size of nanomaterials, as well as zeta potential and the microreology of the solution, particularly useful in proteins and electrophoretic mobility of proteins and polymer solutions. We also have an ICP-MS, which allows simultaneous analysis of multiple elements, and we can analyze trace levels at the ppb level. The elemental analyzer, which allows us to determine the percent composition of carbon, hydrogen, nitrogen, sulfur, and or the presence of chlorine. And this one is useful for characterizing organic compounds. The chromatography laboratory is equipped with the following instruments. The HPLC, which is capable of converting into a GPC or size exclusion chromatography. It is also equipped with the UV and RI detector. And we also have a preparative LC system with fraction collector, which allows us to collect samples from separated components in the LC system. And we also have the LCMS QTOF, okay, which is useful in metabolomic studies as well. And of course, the GCMS, which allows us to monitor components in volatile components in a sample, which is again useful for metabolomic studies. The integrated electron microscopy laboratory is equipped with the following instruments. The transmission electron microscope, or the TEM. So we have an ultra-high TEM, which can do resolution as high as 0.19 nanometer. It's also equipped with energy dispersive spectrometer for elemental analysis, and it can do elemental scanning as well. And along with the TEM, we have an ultra microtome instrument that allows us to thinly slice sample for measuring or for viewing in TEM or SEM. And we have a micro manipulator so that we can prepare samples in the nanoscale. And of course, we have the palladium gold spatter filter, which is useful for preparing the sample. The TEM is capable of analyzing nanomaterials as well as biological samples. We also have the scanning electron microscope, which is also equipped with an energy dispersive EDS, and then it allows us to do elemental mapping as well. Then we have the focus ion beam, which allows us to manage your sample or operate your sample uh, by milling and polishing your nanomaterials, and we can do that in focus ion beam instrument. Upcoming is the imaging and cell culture laboratory, which is ongoing installation now. And we will be having this instrument called the confocal laser microscope with live cell imaging. So this allows us to visualize cells live, and we can monitor the interaction of a compound in interaction with the cell. And this one is also coupled with cell culture laboratories, which allows us to prepare samples from plant cells, mammalian cells, as well as bacterial cells. And of course, for engineering research, we have a wind tunnel facility, which allows us to study aerodynamics and as well as modeling of air flows. So the Central Instrumentation Facility offers programs and projects, and the following. The first one is we offer graduate residency program where we invite graduate students to reside on campus so that they can have access to the instruments 24/7. And we recently graduated two students for this program who did 24/7 access to the instruments and unlimited use of the instruments. Okay? We had Realtech Son, who finished her MS degree last term. Next. And we have Jessa Luteri, who also finished her MS degree last term. And ongoing now, our research resident is Paul Adrian Go, who is ongoing doing his thesis on campus. And we also have the Plus Lunas Fellowship Program, which is funded by PCHRD, where we invite fellows all over the Philippines from different Plus Lunas centers. And we had phase 1 training, which is online, training on chemistry of natural products, NMR spectroscopy for natural products, and chromatography for natural products. So basically, this phase 1 or online session will be followed by phase 2, wherein all these fellows all over the Philippines will be brought to De La Salle University in Laguna campus for hands-on operation of these machines. Then we host Balik Scientists. The first one, as mentioned earlier, was Dr. Leonel Santos. And it was very instrumental in ensuring that our processes are at par with international standards. And of course, right now we are hosting Dr. Ana Karen Lacerna, who will be our keynote speaker for today's session. And the services we offer at the Central Instrumentation Facility, we offer analytical services geared towards your specific objective in your research. We offer trainings upon request. We offer collaboration, and we also do consultation as well as research outsourcing. So for more information about the Central Instrumentation Facility, please contact our general email is cif@dlsu.edu.ph. We also have ongoing construction, the website that contains, so far at least now, the NMR facility. And you can contact us at Facebook and Messenger. Just type DLSU Central Instrumentation Facility. So we hope that through the CIF and our activities, we will be able to help you in your research needs. And please do not hesitate to contact us for your specific objectives of your research. And we hope that we will be able to engage you and in detailed activities on your research, and we hope to elevate the quality and the number of research in the country. And let's help build our country through research. Thank you again, and God bless us all, and may you learn more a lot through this 3-day seminar on TAAS 3. God bless us all.
Thank you, Dr. Camacho, for the introduction to our facility. So sana nainganyo tayo no, sa facilities na pinakita ni Sir, and we hope to collaborate with you in the future. So, again, for inquiries, you may send a message via our FB page, DLSU Central Instrumentation Facility. Type nyo lang yun sa Facebook search bar. And you may send us a message via cif@dlsu.edu.ph. So in this portion, we will now be reintroducing our keynote speaker. She was also with us last week. So our keynote speaker is a Department of Science and Technology, Philippine Council for Health Research and Development, Balik Scientist Awardee. She earned her BS degree in chemistry from the University of the Philippines, Los Baños, and spent her early professional career as a lab analyst at the Analytical Service Lab of UPLB Institute of Chemistry. Later then, she was a quality assurance officer at Me Johnson Nutrition. After which she then spent the next decade in the National University of Singapore as a PhD in chemistry student, research assistant, and as a postdoctoral research fellow, covering a number of projects involving various applications of metabolomics and analytical sciences in areas such as preclinical/clinical, agricultural, food, forensic, and environmental research. In her ongoing tenure as Balik Scientist, she has assisted the CIF through her technical support in the daily operations of the chromatography lab and her involvement as a mentor for the chromatography lab course of the Tuklas Tunas Fellowship Program for Natural Products Phase 1, a project funded by the DOST-PCHRD. Our speaker has also served as a lecturer at the DLSU Chemistry Department, handling the graduate course on special topics in analytical chemistry, specifically food analysis, in Term 2 of AY 2020-2021. The Santa Rosa, Laguna native, a rising research scientist in the field of metabolomics, has published more than 10 papers on the topic, covering various applications. Everyone, we are honored to introduce you to our keynote speaker, Dr. Ana Karen Lacerna.
Okay, thank you very much, Chris, for the kind introduction. So, good morning, everyone. So, we are in our second installment of this metabolomics approach and its different applications webinar series. So, kung hindi po kayo, if you were not able to watch or join us for the first session, please feel free to visit the DLSU Central Instrumentation Facility FB page, and from there you will be able to watch yung first session natin wherein I introduced the metabolomics and its different applications. And we have also heard from Dr. Maria Fe Calingacion on her application of metabolomics in rice research. And for today, we also have great stuff in store for you all. Um, so last session, we were able to address the what, the why, the when, and the where of metabolomics. So you were introduced to the different possible applications of metabolomics. and what metabolomics is. And so for today, we'll be covering more on the how. So for this particular session, we'll cover more of the experimental workflow as well as the practical considerations. And in the next session on March 19, we will be covering more of the chemometrics in metabolomics. So how are the data analyzed in metabolomics. Okay, so again, today we'll cover the house of metabolomics. Um, and we begin by showing you ano ba yung metabolomics workflow that is involved in a particular metabolomic study. So first, you have to define the problem. Second, you have to set up your experimental design. Next, of course, you will have sample collection and subsequent sample preparation, and then data acquisition. And after you have obtained the data, you will have to process the data and analyze using statistical techniques. So dito papasok yung tinatawag natin na chemometric techniques for data analysis. Subsequently, after you have done your statistical analysis and identified the features or variables of interest, you will do your metabolite identification based on the information that you have from the instrumental um, instrumental platforms that you have used. And also you can analyze determine the different pathways that are perturbed using pathway analysis softwares or functionalities. And lastly, it is paramount, or it is of great importance also to work on validations of your results from your initial metabolomics workflow. So dito papasok yung first yung mga statistical validations mo, as well as yung validations of the experiment, making use of independent um, subjects or independent sample set. Okay, so we'll go through the details of this metabolomics workflow, and we'll show you more of like practical considerations and the things that you need to take note for each step. So first, the definition of the problem. So tatanungin nga natin, what is your problem? So I have had discussions with possible collaborators, students before, and they will just tell me, okay, I want to do a metabolomics project. And then, okay, so ano bang metabolomics project do you want to do? So we need to go further to wanting to do metabolomics. Kasi as we've stressed last week, metabolomics is mainly a tool. It is an analytical approach. And so it is directed towards something, towards a particular question that has to be addressed. Kahit sabihin natin na, okay, metabolomics, method development, that is still also directed towards a particular group of analyte that you really want to analyze. So hindi siya yung endpoint. It is just a means to the end. And so you need to identify your scientific question. So what is the scientific question that you want to address? So from the initial question, so okay, anong gusto mong gawin? If somebody says, okay, I want to do a metabolomics project. So next part, anong sample? What is the sample of interest? What is your subject of interest? And then subsequently, you have to ask what is the scientific question that you would want to address, and why is it relevant. So kapag sinabi natin, for example, okay, I want to analyze talaba, for example. Okay, anong scientific question they want to address regarding talaba? Is it yung looking at the quality or assessing the quality of talaba? Is it assessing the possible um, uh, effects of the environmental exposures of the talaba in the different areas that it is grown, etc. And bakit relevant to? So why is it relevant? So dito papasok, ano yung reason why you are looking into these aspects? And then lastly, you also have to ask, can metabolomics provide the answer to your question? So is the metabolomics approach the one or the approach that can give you the answer? And also, ito lang ba yung means to answer the question? Or ito lang, is it sufficient enough? So in some cases, as we've pointed out also last week, it might not be enough to just look at the metabolomics uh, profile. In some cases, you might need to also look at the other omics levels or other possible markers or molecules of interest in order to get the big picture or the clear picture of your study. And I won't go into so much details on this because next to me, we will be hearing from Dr. Mel Garcia of the LaSalle Food and Water Institute. So yung topic niya, metabolomics is the solution, but what is the problem? This is very relevant to this particular part of our discussion. So I'll leave it up to him later to further delve into this definition of the problem in his own applications sa coffee. So, yun. We're looking forward to hearing from Dr. Mel. Okay. So, from the definition of the problem, you go to the experimental design. So, importante na ma-define mo yung problem kasi dito magbabase yung experimental design mo. So, in the experimental design, of course, you have to determine ano ba yung experimental setup mo. Then what is the sample size that you are looking into? For metabolomic studies, you need at least 3 biological replicates. So please take note, hindi technical replicates, hindi instrumental replicates, hindi repeated injections of the same sample, but it has to be experimental or biological replicates. So at least 3. Pero if you can have 5, 6, or even more, that will be much better because in the subsequent chemometric analysis, the more the samples that you have that can represent your metabolic profile, then the more robust your models will be. You also need to define your sample group. So dito napapasok yung mga control and treated groups mo. And if you're looking at longitudinal analysis, then you will have samples of your different groups that has to be analyzed or collected samples from at different time points. So you need to consider that. You need to determine kung ano bang approach yung gagamitin mo. Is it targeted or untargeted? So we'll define this later on. And you also need to determine anong type of quantitation. Is it or what type of analysis will this be? Is it quantitative analysis or more of a qualitative analysis or a semi-quantitative analysis? So more of relative quantitation lang. So you need to define that together with determining whether you are taking your targeted or untargeted approach. You also need to consider ano ba yung kailangan mong instrumentation for this particular in order to address the particular question that you have. So in some cases, if you're looking more at volatile components that are emitting or that are coming out from the biological system, then of course, the more suitable equipment for that will be your Gcms. If you're looking at non-volatile or semi-volatile and thermally labile compounds, then of course you will have you can make use of either NMR and LCM or LCMS, and actually pwede rin naman yung GCMS with derivatization, but these are things that you need to consider. In some cases, you might need to make use of 2 or more techniques in order to give you the comprehensive profile in case you need a comprehensive profiling of your sample. Okay, so last week we have defined yung metabolomics. So we have talked about metabolomics. The term metabolome, again, was first coined by Oliver et al. When they measure the changes in the metabolite concentrations with deletion or overexpression of a gene. On yung metabolomics naman was first coined by Oliver Finn when he talked about it being the comprehensive analysis of all metabolites in a particular biological system, and it involves identification of these metabolites and quantification. Subsequently, actually, yun, medyo mas nauna pala sila. Yung metabonomics was coined by Nicholson et al. When they described metabonomics as the comprehensive and simultaneous systematic profiling of multiple metabolite levels and their systematic and temporal changes caused by factors such as diet, lifestyle, environment, genetic effects, and pharmaceutical effects, both beneficial and adverse in whole organisms. So, okay, medyo mouthful yung definition ng metabonomics, pero basically, metabolomics and metabonomics have been subsequently interchanged, and currently more of yung metabolomics na lang yung ginagamit natin. So, to just summarize, metabolomics involves the simultaneous and comprehensive analysis of metabolites in a particular biological system under study. Other terms that you have to also take note will be, of course, your target. We talked earlier about targeted and untargeted. So we have your targeted metabolomics, wherein you are looking at the quantitative and semi-quantitative analysis of a few metabolites that might be of the same class or belong to the same or linked metabolic pathway. So this is used if you have specific pathways of interest. So for example, if you're looking at the cysteine methionine metabolic pathway or the tricarboxylic acid pathway, then you can do targeted analysis of the metabolites in those particular pathways. Kapag naman ang targeted metabolomics, we are referring to the qualitative or semi-quantitative analysis of the largest possible number of metabolites from a diversity of chemical and biological classes in a given biological system. So, ito naman, it's comprehensive. It's trying to encompass as much of the metabolic profile as possible. So it's trying to encompass as much of the metabolites that are present in the biological system as possible. You also have what is the term metabolic fingerprinting. This is the untargeted analysis of intracellular metabolites. So looking more at cell cultures or in vitro models, analyzing the contents of the cell after a particular treatment or looking at a particular disease state, using a targeted approach is what we know as metabolic fingerprinting. On the other hand naman, metabolic footprinting is analyzing yung extracellular metabolites that are released from the cells into the culture media. So, we expect that if the cells are given certain treatments, they will release some of these metabolites either because of lysis or even because of if they want to release certain signaling molecules. So we measure these extracellular metabolites as well from that are present in the culture media. So yun naman yung metabolic footprinting. And so this is just to show you an overview, like a diagram or visualization of these different approaches in metabolomics. So if you have a cellular or a biological system, if you're looking at the metabolome as a whole, that is which is the internal, the intracellular contents or intracellular metabolites of your biological system, that is known as fingerprinting. And this again, of course, is an untargeted metabolomics approach. If you're looking at what is released into the culture media, or what are the secreted metabolites. This will be your footprinting. And again, this is an untargeted metabolomics approach. On the other hand, if you're looking at a specific class of compounds, like for example, cholesterol, fatty acids, and lipids, or if you're looking at specific pathways, the metabolites of specific pathways, then that is considered as metabolic profiling. So you are looking at the quantitation of specific uh, metabolites or classes of metabolites. And this is considered to be a targeted metabolomics approach. Okay, so it boils down to, in this analogy, do you want to see particular features? Do you want to see a particular, in this case, particular group of buildings of a given type of architecture, or do you want to see the whole scenery? So, do you want to see the whole or comprehensive overview of your system? So, meron tayong approach for either cases. Now, from the experimental design, we move to sample preparation. So ano ba yung mga considerations in terms of sample preparations, and what are the things that we need to um, uh, do or take note of when we're doing the sample preparation, and as well as your initial collection. So hindi natin mo cover everything exhaustively, but we'll give you at least the basics or the fundamentals in terms of like the sample preparation as well as the data acquisition. Okay, so experiment and sample collection. First, you need to do fast collection and stop the metabolism immediately. Kasi in metabolomics, we wanted to capture the metabolic profile or the metabolome at the point of interest as much as possible, di ba? So you have to perform what we call quenching, which we will be explaining later. And then you have to consider also, especially with plants, right? If you are collecting plants, there can be wounding as you cut, like for example, leaves, or if you cut twigs, there will be wounding during collection, and this will result in some metabolic response. And you would want to arrest these wounding responses as fast as possible. You also need to consider to minimize the sources of variations. So such as in sample collections, yung different possible sources of variations in sample collection, such as in if you're collecting samples, like again for plants, if they are of different developmental stages, you will want to stick with one developmental stage of like the plants. Uh, you also need to consider light period and time of collection. And even for biological samples, like for biofluids, in the case of like human and animal models, you would expect the urinal or cycle-based changes in the metabolic profile. So you have to stick to a specific timing in terms of collection. You also need to consider yung harvest duration. So yung harvest duration naman, this will be again for your plants. At what point or at what seasons are you collecting the samples for your study? In some cases, it might be advisable to, if your crops or your samples are being grown throughout the year, then it might be good to like sample at different times of the year in order to obtain a more accurate profile of your samples. Next is sample handling. So in sample handling naman, you might need to consider how the transport, the storage of your samples as well. how they are collected from the site of the experiment, how they are stored, and how they are transported, and how they are taken into the lab. So these are different parts of the process that can also have some effect in your metabolic profile. So you need to minimize the sources of variations in these different steps. So, as mentioned earlier, it is important to capture the snapshot of the metabolism of your sample at the time of collection. Just like in this picture, you kind of like freeze in time yung mga droplets that are present on the twigs or on the flowers itself. The same goes for in metabolism, in metabolomic studies. You need to like capture uh the snapshot, freeze in time the metabolic profile of your sample. And actually, it can involve some freezing, very deep freezing. So when we talk about quenching, again, this is to stop the metabolic activity of the sample as soon as possible from the time of collection. So usually it makes use, we make use of like liquid nitrogen to do this. So yun talaga, if freeze mo talaga yung samples mo in order to stop the metabolism. So, but I understand in some cases liquid nitrogen might not be readily available in some labs, and so you can consider also other quenching techniques. Um, of course, you can do oven or microwave drying in order to dehydrate your samples, and with that, it will also help stop the metabolic activities present. Although you have to take note that this is not suitable for thermally labile and volatile compounds. So if you're looking at thermally labile and volatile compounds that you're subsequently analyzing, um, please do not use oven or microwave drying. You might need to consider other quenching techniques. You can also consider the addition of cold solvent. So this is something that I usually do in my studies previously, particularly if I'm working with biological samples like biofluids. With the addition of solvents like with the addition of certain percentage of solvents like methanol, so if you make yung solvent composition mo to 80% together with your sample, your proteins will precipitate out. And this will stop the metabolic activities right away kasi magprecipitate out na yung mga enzymes mo who are involved in the catalysis of these different metabolic activities. So, that is a common approach, protein precipitation using methanol or extraction, and with methanol chloroform or methanol with methyl tert butyl ether together with water, etc. So usually cold solvent, so it can be stored first in -20 or -40, and then subsequently you use that cold solvent for the protein precipitation. In some cases, if the above cannot be done on site, especially if you're collecting from like yung sa mga sampling site na medyo malayo from your laboratory, cold storage can help slow down the metabolism until access to the quenching methods is available. So if you can keep your samples in cold storage, like in ice box, uh, in its transport to your lab, then that can help prevent uh, that can slow down the metabolism. Hindi siya totally stop, but that can slow down the metabolism. And with this, it is very important to have controls, like a proper control that will go through the same process in order to still be able to identify ano ba yung mga effects that are really relevant to your scientific question. Okay, so this is just an example of, like, in the case of melon samples, for this particular illustration, ano ba yung mga critical steps for the harvest, sampling, storage, and transport of melon samples that are prepared for analysis using different techniques. So in this case, they analyzed yung melon samples using NMR, Gcms, and LCMS. So, as we have shown earlier, very important at the start to establish your experimental design. And then from there, dito pinakita um, how they have determined kung okay, you culture your crops either in the field or in the greenhouse. Subsequently, you have to harvest and you need to take note ano ba yung mga harvest stage and conditions. Um, you need to also, because at this point nga, at the harvest stage, when you are collecting samples, when you are collecting twigs, leaves, etc., the wounding can definitely contribute to the metabolic changes. So you need to, like, limit this or stop these types of changes as early as possible. And here, looking at smaller parts of the plant, then you can do in situ sampling, do organ deep freezing if you can bring some liquid nitrogen on site. Um, of course, again, we highlight the need for biological replication, at least minimum of 3. But if you can have 5 or more, then it will be much better. And then subsequently, magkakaroon ka ng transport to the lab of your samples. Sa large organs naman, sa large parts of the plants, you might have to do yung cleaning, sampling, and biological replication in the lab. You can do your tissue dissection and tissue deep freezing. And subsequently, all samples would be better stored in -80 degrees. So, mas na preserve yung metabolic profile if they are stored in very low temperatures in deep freezing, such as in -80 degrees freezer. Subsequently, you can do your milling, your size reduction. So, kasi if you're going to do subsequent extraction with solvents, it is good to reduce yung particle sizes of your sample in order to increase the surface area and to make your samples homogeneous. So, you can do milling or grinding with liquid nitrogen. And this ground sample can again be stored in -80 as much as possible. Please, you have to avoid. So kung yung milling mo is in liquid nitrogen, subsequently, ipasok agad siya sa -80 para hindi magto yung sample. Because once that the samples get thawed, then that's like one freeze thaw cycle. So in yung repeated freeze thaw cycles can actually also affect the metabolome profile. So we have to limit that as well. So yun, as mentioned dito, as much as possible, no sample towing. Do your aliquoting before the final storage dun sa -80. So if you can do it at this point or right after milling and trying to keep everything still very cold, then much better. Kapag kasi, for example, if you didn't aliquot right away for the different analyses, and then you will, like, take the samples in and out of the freezer in order to get your samples for different analysis, then that will you can expect that there will be metabolic changes already that is not associated with your initial analytical uh, settings or in from your initial analytical profiling. So yun, you have to consider these aspects when we're doing a metabolomic study. Okay, so this is again an example using plant materials. So basically, after you have harvested your samples, you have identified, are you looking at the leaves, the twigs, the roots, etc. And then you have your actual or specific parts of the plants. You can grind them under liquid nitrogen using mortar and pestle. Medyo mahirap siyang gawin if you're dealing with hundreds or like tens to hundreds of samples. So an alternative to mortar and pestle grinding will be, of course, if you have your cryo grinders or like your bead beaters. So that will be an alternative. Um, uh, then you are going to transfer specific amounts or specific masses of your ground samples into a vial or a tube. You are going to have your solvent in there, your cold solvent. It can be methanol or methanol water or methanol water chloroform. So it depends on uh, on your uh, on what you want to extract. So yun. Kasi if you do water methanol chloroform or water methanol methyl tert butyl ether, that will be what we call a triphasic extraction technique, wherein you will have three phases. So you will have the phase of your um, aqueous extract, you will have the organic extract, and you will have your residues. So which will be your cell debris or plant debris, as well as your proteins that have precipitated out. So these will be in contrast to like the more simpler biphasic or two-phase extraction, where you only have like for example 80% methanol. So you only have the extract layer and yung cell debris and protein residues. So these samples can be uh, okay. So this part actually is more for storage, but I apologize for the confusion. So itong part na to, yung C and D are more for like initial storage in minus 80 degrees. So from after grinding, you transfer them to tubes and then keep them in the minus 80. You can do freeze drying to better preserve your samples. So again, this will be to dehydrate your samples, you take out the water and make your samples more stable. And eto na, yung sinasabi ko na you can weigh a certain amount of your samples into the tubes and subject it to extraction using yung solvent. So it can again be yung biphasic or triphasic extraction techniques. So again, that will be dependent on what you want to cover in terms of the metabolome. A very common way of doing it is you add again the solvent into the weighed material, and subsequently it undergoes sonication. So you can do sonication. So yung mga ultrasonic or ultrasonic bath that we use for cleaning our some of our small devices or equipments, it can be very much used for purposes of like extraction. Kasi when we do ultrasonication, nagkakaroon ka ng cavitation within the plant materials or within your samples. And this cavitation, these small bubbles actually will be having high pressure and temperature within them that can disrupt yung mga cells and allow for the extraction or the release of your metabolites into the solution. Um, and yun. So it is a common way. Other methods, of course, if you have microwave extraction devices, or if you can even do just vortex mixing for a certain period of time. But again, that will be a bit tedious if you're doing it manually. So it would be good to have like some automation in place. But yeah, for me, um, I usually make use of ultrasonication in my when I'm extracting um, tissue samples. Okay, so one consideration also, if you're doing sonication, please take note, the temperature dun sa sonication bath mo will increase kasi nga there is some heating involved as the sonication process goes or progresses. So if you want to keep your metabolites um, if you want to keep your samples cool, then it's good to also incorporate an ice bath para rin makatulong na hindi masyadong mag-overheat or hindi masyadong ma-heat up or yung samples mo. Okay. So subsequently, you can also consider kung hindi man sonication, we also make use of those ball mill or bead milling. So you have your sample added to a tube which contains zirconium oxide beads or silica beads. And then this will be put into equipment that will be shaking at very high RPMs, and this will yung presence ng mga beads doon, yung mga collisions of the beads with your sample can actually break your samples and extract and allow the extraction together in the presence of the solvent. So this is another approach that you can take in terms of like the extraction. Subsequently, if once you're done with the extraction, usually we keep the samples in cold temperatures, either sa fridge or sa freezer for a certain period of time to aid also in the protein precipitation. Kasi di ba kapag low temperature, solubility will decrease, and in this case, this will actually help in the precipitation of your protein. So you can keep your samples in ice or in -20 degrees for about 10 minutes to just help in the precipitation of your proteins. Subsequently, you can separate out your supernatant using centrifugation. So usually you can set it at high speed in order to separate out your cell debris and residues and protein precipitates from your supernatant. So usually around 14,000 RPM in a small benchtop centrifuge. If you have refrigerated centrifuge, that will be much better so that you ensure na hindi nag-heat up yung samples mo as it undergoes centrifugation. So, these metabolites are of course stable kasi yung normal body temperature naman natin is at 37 degrees. So, anything below that will be good. Kapag more than that, medyo we might expect like some possible effects in terms of the metabolites. So yun. So if you have refrigerated centrifuge, much better. Kung wala naman, then you might consider having to decrease the time that you do the centrifugation. So pwede na decrease the time and then do it twice. Pero meron kang parang cool down phase in the middle. So that's one approach to that. And then once you have the supernatant, in this case, it was subjected to NMR analysis. So right away from the supernatant, you can transfer it to an NMR tube and subject it to NMR measurements. For LCMS analysis, the same can be done. Yung centrifugation mo can help to can already help separate out small particles from your extract, and with that, pwede mong directly inject sa LCMS system mo, but there can still be risks of clogging of your columns. So it's still advisable to make use of your filters. So at least 0.45 micron, or if you have 0.2 micron filters before you do before you inject the samples into the LCMS. Yung sa GCMS naman, this one you'll have to see kasi you might still need to do derivatization for your samples before you do your GCMS. So as mentioned here, similar yung workflow up to for MS-based metabolomics up to point I, and then subsequently magkakaiba na lang as it is subjected to either NMR, LCMS, or GCMS analysis. For solvent extract, if solvent extraction is done at the onset and the immediate instrumental analysis cannot be performed, the supernatant after centrifugation can be obtained and dried before the freezer storage. Kasi in some cases, for example, if you're looking at longitudinal analysis subjected to LCMS or GCMS analysis, kasi yung GCMS and LCMS can be prone to batch effects. So if you analyze your samples at different times, makikita mo na possible na yung nakikita mo differentiation of your samples might be due just to effects of the difference in time in analysis. So in order to avoid that, it is advisable to analyze your samples at the same time. So ang ginagawa namin usually, especially for looking at large cohort studies, we do the extraction in batches wherein hindi kami nagsastop at taking at the supernatant. So after taking the supernatant, we subject the sample or the extract to drying. Pwedeng drying using nitrogen gas or drying using using vacuum centrifugation or speed vacuum centrifugation. If your sample is mainly an aqueous extract, you can use freeze dryer. So once that is in dried form, it is again stable and can be kept in the -84 storage until it is time for you to do the analysis. So that's an approach that we also usually take when we're dealing with large amount of samples. Okay, in terms naman ng biofluids, yung mga biological fluids like in this case urine and plasma, serum, as well as other biofluids that is often used in clinical metabolomics like cerebrospinal fluids, sputum. I think right now we're already also seeing applications of metabolomics to saliva, and etc. There's currently almost all the different types of fluids or samples that can be obtained from biological systems are already being subjected to analysis. So dito na rin papasok yung mga tissue samples mo, and yun nga yung mga biofluids, urine, CSF, BALF like bronchoalveolar lavage fluid, sputum, saliva, blood. Of course, that one is a very common sample for clinical metabolomics. Sweat, meron na nag-analyze ng sweat. Si Sir Rod, he's looking at the metabolome of milk. So, very interesting. So yun. So there's a lot of different factors that can also affect these different biofluids. So for this particular session, we'll just give you a glimpse of like what are the factors that can affect the metabolome ng urine at saka ng blood. So in this case, we're going to obtain either plasma or serum. Okay, so as you can see from the different stages ng sample preparation prior to your metabolomic analysis, there's a lot of steps there are that can be, and there are different factors that can be affected in these different steps. So looking first at your blood, so yung choice of collection tube, certain collection tubes will have like different coatings, and this can affect your metabolites. Either they can be like they can be capturing your metabolites from your solution, or they can be a source of contamination for in the subsequent steps. If you're looking at plasma, so distinction natin plasma and serum. So serum is yung component of the blood that is obtained after it is allowed to coagulate. So after ito yung ina-allow natin mag-coagulate yung supernatant. So initially, you will subject your blood sample to centrifugation. Of course, you will have your cells in the residue, and then you have a supernatant, and this supernatant can be subjected to if it is allowed to coagulate, so separate mo yung supernatant from your coagulating factors or other cells, and other components, and yung serum will be the supernatant. On the other hand naman, kapag hindi mo inallow na mag-coagulate um, yung yung component na yon, yung supernatant na yon, and it is put in collection tubes that is coated with certain anticoagulants like citrate, heparin, EDTA. So, ang tawag naman doon is plasma. So, yun. So, if it's not coagulated, plasma yun na obtain natin. So, again, this choice of like which anticoagulant that is used, they can also affect yung metabolic profile. So, actually, yung heparin, EDTA, they can be used. Yung citrate, medyo may problema tayo doon kasi citrate is a metabolite. And so if you're looking at metabolites that are like, for example, citric acid and related metabolites, then yung levels of your citrate in your anticoagulant can be a problem kasi yun nga, they're the same. They are actually present in your metabolome profile. You also need to consider ano ba yung sampling device. Is it a vacuum sampling device or aspirated? Ano ba yung sampling site? Is it a venous blood, or is it collected from the artery? In terms naman ng sample processing, yung clotting time for the serum is an important factor. Of course, if there is differences in clotting time, and actually for different individuals, the clotting time can be different as well. So this can be a source of variation. Centrifugation, of course, if you subject kasi yung blood to very high speeds of centrifugation, then this might actually lyse yung cells. So once the cells are lysed, so maapektuhan na nun yung metabolic profile mo nung plasma or nung serum. Kasi kumbaga, hindi na lang siya metabolic profile of your plasma and serum, pero nakasama na rin doon yung profile nung red blood cells mo, for example, or your white blood cells. So you need to be careful with that. I think, if I'm not mistaken, it's 3,000 G for centrifugation for like this initial part of centrifugation if you're trying to separate out your serum or plasma from your the rest of your blood components. And of course, in some cases, there can be hemolysis. So if your samples already has, if there is already hemolysis in your sample, then you might have to consider not including those samples in your study. And yun, time delay and temperature. Again, this your metabolome changes very fast. So I think it will be in the range of hundreds of millimolar or tens of millimolar per second. So you need to be careful to limit as much as possible time delays. And if you can keep your samples in cold storage during this whole time of like collection and processing, that will be much better. That will help slow down the metabolism and preserve your sample. And yun, aliquoting and transport. So again, especially for biofluids, it is good to do your aliquoting before you subject them to long-term storage, such as yung -80 or -20 or 4-degree storage. So for long-term storage, usually you can store it up to about 1 year. You can store in -80 for short-term storage of just a few days to a few weeks, you can keep it in -20 or in 4 degrees. But again, yeah, you have to consider. I think yung urine can be kept in 4 degrees up to about 48 or 72 hours. So that will not be an issue. But yeah, possibly in the case of plasma or serum, it might be better to keep them if they're it might be better to do the cold extraction right away, dry them, and then keep them for long-term storage. Okay, so in the case naman ng urine, collection drugs, again, the additives that are present in the collection drugs will affect it might affect your profile. Sampling modes, you have to consider, are you going to do spot sampling, time 24 hours? So again, this is because there are changes in the metabolic profile based on the at different times of the day. The metabolic profile can be different for a particular person. So um, and then you have to consider, is the sample collected midstream or first void? So as much as possible, collect from the midstream. So mas consistent, mas reproducible yung sample that is obtained from midstream. And sampling time, yon. So again, consideration of like when it is collected. So usually it's advisable to do the collection in the morning, so that will be more reflective of your nutritional I think your nutritional status. And ayun, so kumbaga kasi yung mga activities during the day, your level of dehydration during the day, your level of hydration during the day will be affected. So it is good to like consider collection uh, from morning in terms of like urine. And then again, also centrifugation. You have to consider the speed of centrifugation, as very high speeds can lyse cells that are present in your urine, including yung mga cells that are coming from your bacterial cells. So there can be bacterial cells present in the urine, and this can also lyse and be incorporated into your urine profile. It is good to do filtration. So 0.2 micron filtration will be a good step to help preserve your sample. So remove mo na with 0.2 micron filtration, you can remove most bacteria that might be present in your sample, and you can then subsequently store your samples for longer term. You can consider adding preservatives or bacteriostat agents like boric acid and azide, sodium azide. But if you can do without them, it's actually also better, especially with boric acid, because that can affect your pH, of course. And then yon, when we talk about same thing, it can be stored long term in -80, short term in -20 or 4 degrees. Um, Towing, again, as much as possible, limit freeze thaw cycles. Okay, so these are just some recommendations for the standardizing of the pre-analytical phase in blood and urinary metabolomics. So they have suggestions. You can check out this paper. So they are the ones who presented those pre-analytical factors that can be affected, and these are their recommendations in terms of like what you can do in the different steps. So in sample collection, so as mentioned here, yung ano ba yung suitable na anticoagulant. As mentioned, citrate tube should be avoided. Sampling time. So all samples must be collected within the same time lapse under the similar fasting status and other confounding conditions. So as much as possible, with limited physical activity muna. Kasi any physical activity can actually affect also the metabolic profile of your subjects. And yun, sampling material, kung anong ginamit mo with regards to the sampling material, it's advisable to make use of the same brand and same brand or manufacturer of your sampling devices, collection devices throughout the entire study kasi the minor change in brand of like centrifuge vials or your collection vials can actually affect the profile of your samples. Also, as mentioned here, yung mga aspects such as yung effects ng centrifugation and or filtration. So, as much as possible, avoid high speed centrifugation if you're trying to separate out your plasma or urine or serum samples from your cells. Kasi nga, lysis can happen if you do high speed centrifugation. As much as possible here, as mentioned, try not to use preservatives in the blood because again, there might be effects in your metabolic profile. For urine, you can make use of sodium azide or boric acid, but more advisable sodium azide. Quenching, you can do snap freezing in liquid nitrogen but not dry ice. This is for liquid samples kasi dry ice is carbon dioxide, di ba? So it can solubilize in your samples, and once it's solubilized in your samples, it can affect the pH of your samples. So you have to take note of that as well. Yun, sample aliquoting. So it's advised to do sample aliquoting before uh, keeping it for long-term storage in order to avoid the frequent freeze thawing. So if you already know how much samples do you need per type of analysis or per type of extraction, so you can keep aliquots of that in your small vials already. And subsequently, once you are ready to do your extraction, so kukuha ka lang dun. Okay. In terms of sample transport, so samples must be transported after pre-processing, aliquoting, and freezing whenever possible. So cold temperature shipment is definitely better or is strongly recommended in order to avoid yung mga metabolic conversions. Sample storage, ayun, so up to minus for 1 week up to minus 20 degrees. Long term, of course, will be using your minus 80. Yun, try to avoid sample freeze thawing cycles. So limit yung freeze thaw cycles. Okay, so that is for your sample prep. You can also, in the case of, okay, in terms of untargeted metabolomics, we don't usually perform so much of these cleanup steps kasi nga we try to cover as much of the metabolome as possible. In some cases, these cleanup steps can actually capture some of the metabolites of interest. So, medyo limited yung cleanup steps when we're doing untargeted. But if you're doing targeted analysis, you want to focus on specific compounds of interest or like even exogenous metabolites, yung mga metabolites ng mga drug treatments mo or pollutant exposures mo. So you might consider doing some cleanup in order to enrich your sample and to remove other interferences. So yun. So you can make use of solid phase extraction or solid phase microextraction. So this one will be a smaller scale of your solid phase extraction, yes. And this can help pre-concentrate or enrich your sample prior to analysis. So there are different modes. So it can be fiber, sample flow based, vessel walls can be coated with the sorbent or adsorbent material. It can also be suspended on particles, so it can be coated on suspended particles. It can be on the stirrer. So a development of this will be your stir bar microextraction, stir bar sorptive microextraction technique. It can also be on disc or membrane. It can also be for volatile organic compounds. It can also be used in the headspace. So you have your headspace SPME setup for the capture of volatile organic compounds for your samples, and subsequently, this can be directly injected into the GC injector port, where there will be desorption of your volatile organic compounds and subjected to GCMS analysis. Okay. Next, after the sample preparation, we go to data acquisition. So data acquisition, dito na papasok yung mga instruments na ginagamit natin for analysis. So this one I have shown you previously. So in metabolomics, these are the different instrumentation that we usually use. So you have mass spectrometry based techniques. You have NMR spectroscopy, and these spectroscopy, your IR, NIR, and Raman, are commonly used for purposes of metabolic or metabolite fingerprinting. Pero in terms of like being able to determine kung ano ba talaga yung metabolites that are changing and what are the pathways affected, you need to be able to know ano yung metabolites. And for that, yung NMR and MS-based techniques will be much more suitable as compared to using yung mga IR, NIR, and Raman. So under MS-based, you can do direct injection or flow injection, or currently you have your ambient ionization or yung mga direct ionization modes in terms of like mass spec. So these are also other options. You also have your couplings with different separation techniques such as LC, GC, and CE. And we'll try to discuss more on yung LC and GC later. Okay, so there is no single instrument that can cover the whole metabolome. For example, mostly if you talk about GCMS analysis, ang mako-cover ng GCMS will be about 30 to around 30% of the small molecules. Kasi nga, you have to consider that in GCMS analysis, it mainly covers thermally stable as well as volatile components. Of course, there are configurations that can help you analyze thermally labile components and non-volatile components. Of course, you can do derivatization in order to allow you to analyze some of the non-volatile components, but there are still some limitations to that. So, on the other hand naman, yung yung LCMS can mostly cover more of your non-volatile, semi-volatile components, and they are more suited for thermally labile. Yung NMR, it can cover a whole range of different metabolites, pero meron kang konting disadvantage in terms of like how sensitivity is, kasi medyo mas mababa yung sensitivity ng NMR compared to like your mass spec techniques. So meron siya kanya-kanyang advantages and disadvantages. And so no single instrument can actually cover the whole metabolome, and you would usually need to use complementary platforms to maximize the metabolite coverage. So for example, if you can do a combination of LCMS, GCMS, you can do NMR, LCMS, you can do NMR, GCMS. So depende dun sa coverage that you want. In my application, previously, we made use of NMR for the more polar metabolites. And actually also, dun sa LCMS side, we made use of two different separation techniques, yung reverse phase and hydrophilic interaction chromatography, to allow us to cover both yung mga polar as well as yung mga non-polar and mid-polar components of the samples. Now, okay, so this is just, I just wanted to use this illustration to show you na, okay, if you're taking pictures during the F1 race, so if you do not have the right gear and the right settings, ito yung makukuha mo. Whereas if you have the right gear, the right settings, the proper, you're aware of how to shoot this properly, then you would have an amazing picture like this. So, importante that you make use of the right equipment, you make use of the right settings, and you have sufficient know-how in order to capture the snapshot of your metabolic profile. So niliken ko dito kasi syempre yun nga mabilis yung changes in the metabolic profile, and so you have to really have the right gear, the right settings, the proper know-how in order to really capture your metabolic profile accurately. So, differentiating your NMR and MS-based metabolomics. So, ano ba yung advantages, disadvantages of NMR, LCMS, and GCMS for metabolomic analysis? So, in terms of sample preparation, medyo mas madali yung NMR kasi you only need to dilute your sample. For liquid samples, you only need to dilute them with deuterated solvent. For solid samples, there will be some extraction involved. But again, if you do it with deuterated solvent directly, pwede mo nang isubject yung supernatant to analysis. Sample preparation for GCMS, if you do headspace analysis, you don't need to do much kasi you can just put your samples in a vial, and then merong heated incubation naman dun sa instrument if you have a headspace analysis module. And from there, it can already obtain your volatile components, so direct na. In some cases, you also need to do extraction with your volatile organic solvents, and yun yung i-inject into your GC. And yun, in order to analyze relatively non-volatile components to increase your coverage, you might need to do derivatization. So dito papasok yung derivatization mo with BSTFA to in order to silylate your sample, or the use of fatty acid, derivatization of your fatty acids to methyl esters, etc. In LCMS, you might need to do desalting and filtration. And yun, extraction with organic solvents. Sensitivity, as I've mentioned, medyo at a disadvantage ang NMR. Unless, of course, I think if you make use of cryogenic traps or cold traps, then that might increase the sensitivity. But relatively compared to your LCMS and GCMS, medyo mas less sensitive siya. So yung detection limits is only up to the micromolar to millimolar range, whereas MS-based will be picomolar to micromolar. Quantification, GCMS and LCMS, kailangan mo ng mga standards, whereas yung sa NMR, hindi kailangan. So you can make use of your internal standards that you incorporate, such as yung mga TMS, TSP, or other internal standards, and these can be directly used for quantitation kasi linear yung response. And in terms naman reproducibility, as I mentioned, GCMS, LCMS can be subject to batch effects, whereas yung NMR has very high reproducibility, hindi siya usually affected if you analyze your samples in different batches or different days. Runtime, usually greater than 10 minutes for both LCMS and GCMS, and this can extend up to about an hour. NMR usually takes about 5 to 20 minutes. The cost for analysis is usually higher also for GCMS and LCMS kasi relatively instrumentation and maintenance costs also is higher for both as compared to NMR. Although you have to factor in yung mga refills of the cryogen for NMR, pero in terms of the actual number of time for usage, as you can see, medyo mas mababa siya. So less yung time that you need to pay for if you're doing per hour charging or yun. In terms of like tissue samples, yun nga, depende sa compounds that you're looking into. You might require derivatization for GCMS. With LCMS, kailangan din extraction. NMR, most likely kailangan din extraction, but if you have set up for magic angle spinning NMR, then you can do direct analysis. Specific advantages for GCMS will be the sensitivity and the availability of standard library. So yung mga NIST library mo, from where you can, which you can use to identify your compounds, and the NIST library has thousands of compounds that you can use for identification. So in terms kasi ng ionization ng GCMS, it's very reproducible, particularly yung electron ionization. So the NIST libraries and most of the libraries will be based on electron ionization at 70 electron volts. And definitely these can be comparable across different instruments. And so pwede mong gamitin yung libraries to determine the identity of your compounds. O sa LCMS naman, also sensitivity is an advantage, selectivity and specificity of detection also, and the higher number of detectable molecules kasi nga LCMS compared to GCMS in terms of the molecule coverage, medyo mas madami siya. There is no need for derivatization. On the case naman ng NMR, it is a non-destructive sampling technique. It can provide you good replication, high reproducibility, and it gives you structure information. Although, of course, LCMS, GCMS will also provide you with some structure information based on its mass to mass, Msmx fragmentation patterns. Specific disadvantages for your GCMS, of course, in some cases, the derivatization can be tedious. It is a destructive detection technique. It is not suitable for thermally labile and non-volatile compounds. Of course, again, there can be configurations or modifications in your method that can be arranged to allow to further expand your coverage. Pero yun, medyo may additional configurations needed. And for both for any mass spec based techniques, your compound has to be ionizable. So it's either it has to be inherently ionizable or you convert it into something that is ionizable, again, using derivatization. So on the other hand naman, in terms of NMR, one of the concerns nga is the low sensitivity. And if you are looking at specific classes of compounds, for example, sugars, there can be signals that are highly overlapped. So medyo mahirap na siyang i-distinguish. You might have to resort to like 2D NMR in order to like distinguish them. But it's a very useful tool if you want to get an overview of your metabolites that are present in the sample. So this is, again, in Nmr Metabolomics, actually, the proton is the nuclei of choice for metabolomics. Although you can make use of your phosphate, your carbon, your nitrogen, uh, pero kasi in terms of abundance and ubiquity, mas preferred yung proton kasi with that you can get high um, intensity signals even if you don't make use of too many scans. If you make use of carbon-13, the carbon-13 abundance, natural abundance is low. Then you will have to use very high concentrations of your samples as well as higher number of scans in order to get a good spectra. So sa proton, walang problema. The metabolites can be identified from database or literature based on chemical shift and multiplicity. So this is an example of the proton NMR spectra of green tea. So yung green tea that is extracted in DSS, and you can see that you will be able to identify a whole range of different metabolites that are present in your sample. So as mentioned, it can provide you a bird's eye view of the different molecules that are present in the sample. Here, we're showing magic angle spinning, NMR. So ito, sinasabi ko that you can use it for tissue samples, so direct analysis of your tissue samples. So it allows high resolution NMR analysis of intact tissues. So this is important if you wanted to study the compartmentalization of the metabolic profiles in tissue. Kasi di ba, for example, in kidney, so meron kang iba't ibang different types of cells that are present in different parts of your kidney. And so if you want to really really profile specifically these different parts, then magic angle spinning NMR will be a good approach to these types of studies. So yun, since magic angle, so it makes use of the magic angle of 54.7 degrees in terms of the tilt of the sample. And this reduces the line broadening effects that are usually present when you're analyzing tissue samples. So this is quite straightforward. So you just put your sample inside this tube. So about 1 millimeter cube of sample, and then you add a bit of deuterated solvent, so about 10 microliter of solvent, and then ayun, so you have to put it inside the probe, and then you can detect your different metabolites. So this is an example of the analysis of a pig liver tissue that is analyzed using a 600 megahertz NMR with magic angle spinning. So you can see the profile of different groups of or classes of metabolites using this approach that is present in your pig liver tissue. In terms of mass spectrometry instrumentation, I'll quickly go through this because I'm running out of time. So first, you have the inlet that introduces the sample into your mass spectrometry system, and then your analytes are ionized in the source or in the ionization source, and then they are subsequently subjected to separation or filtering using your mass analyzer. So this is based according to their mass to charge ratio using magnetic or electric field. Um, then your detectors will count the ions that produces the signal of your separated ions. And you, of course, have your instrument control and data system present in your PC for the control of the instrument and the recording of the signals. And this system, the mass spec is usually in under high vacuum. So in some setups, the source might not be under the vacuum anymore. So this will be in the case of your atmospheric pressure ionization. So hindi na siya naka-vacuum, but in other setups, this will still be part of your vacuum chamber. So yun. So okay, so vacuum system, it's important for minimizing ion molecule reactions, scattering, and neutralization of ions. Okay, in terms of choice of ionization technique, there are in GCMS, it will be mainly your EIN, electron ionization and chemical ionization. LCMS will be your APCI, atmospheric pressure photo ionization, APPI and APCI yung chemical ionization counterpart niya, and also electrospray ionization. In MALDI MS, of course, you have your MALDI ionization. So as you can see, in terms of coverage, most coverage can be afforded making use of ESI for LCMS. You have different mass analyzers. So after the ions are formed, the ions are accelerated into the mass analyzer, and they are separated according to their mass to charge ratio. You have two types of analyzers, your continuous analyzer, such as your quadrupole filters and your magnetic sectors, and your pulsed analyzer, such as your time of flight and ion trap. Some important terms that you will usually hear in mass spectrometry. First is resolving power, the ability of a mass spec to separate adjacent ions, and your mass resolution. So this is a measure of your resolving power. So it is measured if you take your peak mass and divide it by the full width at half maximum. So this will be your full width at half maximum. And as you can see, a high resolution MS will have a very small full width at half maximum compared to your quadruple that has a high um, full width at half maximum. And so from here, you can see na kapag sinabi natin high resolution, definitely with the narrower peaks or the narrower signals, you will be able to separate adjacent ions better. Mass error and mass accuracy. So it's a measure of the accuracy of the mass information that is provided by the mass spec versus the true exact mass. You can get the mass error by subtracting your measured mass from the theoretical mass, and your mass accuracy by dividing the mass error with your theoretical mass multiplied by 10 to the 6th. Monoisotopic mass is also known as your exact mass, which is the atomic mass for each um, um, the molecular weight wherein the atomic mass for each atom is based on the highest abundance isotope for that atom. So yun. this is a comparison of the different mass analyzers. So you have your quadrupole, you have ion trap, you have time of flight, magnetic sector, Fourier transform, ion cyclotron, resonance, MS, your orbit trap. So they have different differences in terms of like mass limit, resolution, um, mass accuracy, ion sampling. So you have those that are considered high resolution MS. So that will be your TOF reflectron, magnetic, FTICR, ICR, and orbit trap. Very commonly used in terms of tandem mass spectrometry, or the combination of two mass analyzers for tandem MS will be your triple quadrupole. Of course, there are also combinations of quadrupole TOF or quadrupole trap, etc. In terms of like tandem mass spectrometry also, okay, I'll go into the next slide. So when we talk about tandem MS, it is a process in which the initial mass analyzer selects a parent ion or precursor ion and subject it to fragmentation. And subsequently, the fragments or the product ions are then mass analyzed by a second mass analyzer. So that will be mainly tandem in space. So you have two different mass analyzers. You can also have a tandem in time, which is your ion trap. So yung ion trap kasi, from the term itself, it can trap the ions, and then in that one trap, pwede niya ma-isolate or ma-filter out the rest of the ions, one fragment one ion and subject it for subsequent detection. Kapag sinabing tandem in space, yun nga, kailangan mo ng at least dalawang mass analyzers. So MSMS is the experiment performed using tandem instruments, and they are called Msmx experiments or tandem MS experiments. Product ions will be the fragment ions formed after CID or collision-induced dissociation. You have your parent ion, which is, or your precursor ion, which is the initial ion that is selected by the first mass analyzer, and that which will be subjected for fragmentation. So this is just to illustrate. So from in tandem in space setting, such as in the case of a triple quad or triple quadrupole, initially you will have in the you have a few ions that will be filtered, from which your Ma plus will be filtered, and these Ma plus can be subjected to your fragmentation, and the Q3, your second quadrupole, will allow for the scanning of these different ions. Or in some cases, if you're looking at multiple reaction monitoring, from these different fragments, the second quadrupole can actually um, filter off the others and just specifically send off for detection one of the ions. So Q1, Q2, Q3, kaya siya tinawag na triple quadrupole, but actually quadrupole mass analyzers are only your Q1 and your Q3. Yung Q2 is a collision chamber. This is an example of tandem in time. So in the case of ion trap, so again, if it will initially trap your ions, select a particular ion to stay inside the trap, and then subject it to fragmentation, and these subsequently can be detected. Again, this is just to illustrate the tandem MS in space setting. These are the different acquisitions modes that you can do in mass spec. So you can do scan. So that means all the ions in a particular mass range can be scanned and sent to the detector. You can have selected ion monitoring for single mass analyzers, wherein you can select specific ions that will be subjected for detection. In tandem in space MSMS, you have a bit more different types of acquisition modes. You can have product ion scan. That means a particular ion can be fragmentation, and all resulting ions will be scanned. Selected reaction monitoring, a particular ion is selected for fragmentation, and only specific fragment ions will be detected. So mas mataas yung selectivity niya or specificity. Neutral loss, only those ions producing a particular loss of this structure will be detected. And precursor ion scan is it will select the fragments that are produced from this particular group of um, ions. So usually, yun nga, MS is coupled with mass spec. So it can provide you structural and mass information. So especially in the case of QTOF, for example, you obtain high resolution MS spectra and high accurate mass. So you can make use of the mass information for identification, fragmentation that can be coming from your EI, electron ionization in the GC side, or in your subsequent MSMS for LCMS or GCMS configurations can also provide you with structural informations that are useful for identification. And if you combine these in the case with GCMS with retention time index information, then you can have very reliable identification when you're using your compound library. So this is just to illustrate the three-dimensional nature of GCMS or even LCMS data. Okay, so some requirements for GCMS hyphenation. Of course, you need to make sure that your analytes are volatile and thermally stable and ionizable. I won't go through so much details with the other aspects, but GCMS can be actually directly coupled with MS kasi nga nasa volatile state na yung ions mo. This is just to illustrate the different GCMS components so you can see the direct coupling of your GC with your mass spec. These are some of the mass analyzer configurations that you can have. I've personally used GC QTOF, GC triple quad, as well as the GCMS DC system at CIF. LCMS hyphenation, so it can be analyzed for a diverse range of non-volatile compounds, including those of high molecular weights and polarities. But main requirement is that the eluent and the components of the eluent must be from the LC must be volatile. So this includes your mobile phase and additives. So usually, usually ginagamit yung mga organic solvents like methanol, acetonitrile, and yung mga additives mo will be also volatile, such as your formic acid, acetic acid, ammonium formate, ammonium acetate, etc. So these are some of the instruments that I have worked with in terms of LCMS systems. So this is the HPLC QTOF at CIF. These are my previous babies when I was working in Singapore. This is an HPLC QTOF system as well as a UHPLC QTRAP system. So QTOF is mainly for high resolution mass spec, and this is QTOF or any high resolution mass spectrometry setups is mainly used for untargeted analysis. Of course, they have also capabilities for targeted analysis. There are certain capabilities for that, for selected reaction monitoring, but mainly for um, targeted analysis, it will be good to make use of QTRAP or triple quad kasi meron kang capabilities for multiple reaction monitoring. And this is very helpful if you're dealing with very complex samples such as blood or plasma, etc. So these are just some considerations in terms of like running your LCMS analysis. Usually column diameters are 2.1 to 3 millimeters high throughput. You can employ 30 to 50 millimeter long columns. For complex mixtures, you might need longer columns such as 100 or 150 millimeter length. Again, mobile phase has to be volatile. Flow rates has to be within the limits of the mass spec. And you have to choose the suitable ion source for your analytes of interest as well as the suitable mass spec for your analytes. And the desolvation temperature, it varies with also flow rate. So depende, higher flow rate will require higher desolvation temperature. But you cannot set it too high kasi pwede niyang ma-fragment yung molecules mo, and this is your in-source fragmentation. So this is just to illustrate kung ano yung difference between targeted and untargeted LCMS or GCMS analysis. So again, untargeted mainly exploratory. You make use of high resolution mass spec. Usually you acquire scan acquisition mode, so you can obtain something such as this, wherein you have a whole slew of different peaks or different metabolites present. If you're looking at targeted, you make use of like targeted acquisition mode, such as multiple reaction monitoring, or or if you're looking at specific classes of compounds, you can make use of precursor ion, product ion, or neutral loss scans. This is just to illustrate the workflow that I have used in a previous study. So you can have your plasma sample subject it to protein precipitation, and then from the supernatant, you can get aliquots for both helic LCMS and reverse phase LCMS. If you are doing the analysis at a different time, then you can do the drying of your extracts and then just reconstitute it just before the analysis. The same plasma sample has been subjected to NMR analysis just with the addition of a saline 10% D2O with the internal standard DSS and subjected to 500 megahertz proton NMR. Okay, so some important considerations when working with mass spec-based metabolomics. First is randomization of your samples, analysis of blanks, such as your extraction, reconstitution, and mobile phase in case there are any contaminants in any part of your um, experimental procedure, then you will be able to determine that or get to know of that by the analysis of your blanks. If possible, best to run all samples for an experiment in one continuous run in order to avoid batch effects and make use of quality control samples. So this is how you prepare quality control samples. If you have different study samples, you can pull them together and from there create small aliquots that will be subjected to the same steps of preparation and analysis, such as your samples. This is usually analyzed at the start of your run for equilibration of your column with your matrix. And also, it can be analyzed every 5 or 10 samples to serve as yun nga, quality control. In case anything happens within a particular batch of samples, then you will immediately know from your quality control samples. So that's it for today. I'm sorry, I have like extended quite a lot, but I hope you have learned a lot from this session. We'll talk more about the data processing as well as the subsequent steps in our next session in March 19, Chemometrics in Metabolomics. So again, this is a 3-part seminar. We're looking forward to the presentation of Dr. Mel later. And of course, in the next session, Dr. Rod Sumaya will be joining me for the 3rd installment of this webinar series. So acknowledgement, again, much thanks to the DOST, PSC HRD, and the Balik Scientist Program, the De La Salle University Manila, Prof. Drexel Camacho, the DLSU CIF staff is also very much involved. There are the people behind this webinar series, Dr. Fekaling Ashon, Dr. Mel Garcia, and Dr. Rod Sumaya for joining us in this webinar series. National University Singapore and its chemistry department who provided me with a scholarship when I was in for my PhD. Prof. Sam Lee and this research group members who has been of great support throughout my research journey in Singapore. The NUS Environmental Research Institute staff, researchers, and management who has also been of great help and support throughout my journey. Applications, I would like to give a special shout out to application and product specialists, sales executives, and service engineers of different instrument and consumable manufacturers. I thank you for their generosity in terms of the help, the support, the assistance they have given me in maintaining our instruments, as well as in helping me get my research work done by providing the support and the products that I need. Of course, much thanks to my family and friends for, again, the always ever constant support and encouragement. And again, all praise and thanks be to God for this opportunity. So that's all, folks. If you have any questions, if you want to explore the use of metabolomics in your analyses, My contact details are here, so please do let me know. So that's all again. Thank you.
All right. Wow. That was really a lot. We learned a lot. So wow, thank you, Doc Karen, for the culminating session on the introduction to metabolomics. And as usual, solid content, and we really learned a lot from your experience on the metabolomics workflow. So to recap your talk, we were introduced to the metabolomics workflow, terms to remember, including targeted versus untargeted metabolomics and metabolic fingerprinting, experimental design and sample collection, quenching, critical steps, sample prep of plant materials, factors that can affect the metabolome of biofluids, common analytical platforms where MS-based, NMR, and spectroscopy-based metabolomics were explored. And these techniques were also differentiated, while some examples were also given, including their important considerations. So there was also an in-depth look at mass spec instrumentation, which will be quite useful for a lot of us, especially those who are currently studying instrumental methods of analysis, those who are currently using an MS as a refresher for you, and those who are planning to purchase a mass spec in the future for your research needs. So also, the multi-platform approach was emphasized to cover the weaknesses of other instruments and maximize the quality of data in obtaining the metabolic profile. So fortunately, the CIF has most of this equipment. So should you be interested in performing metabolomics research, don't hesitate to send us a message via FB or email. So again, as Dr. Karen said earlier, next week on Friday, March 19 will be the last session for this webinar series. At 9 a.m., we'll be having chemometrics. So if you're interested in data processing, be sure to attend next Friday's session. So ayun, I'm sure we all want to look back on the contents of the webinar at some point. So don't worry, our streamed content are always available in the DLSU CIF Facebook page live tab. So just get there and you may stream the content, previous content, or even this content later um, anytime you want. So Dr. Nasarna also give a quick patikim of Dr. Garcia's talk later. So let's wait for that. And then shout out muna tayong konti, okay? Shout out to my CIF teammates, Hazel, Alona, Jude, Doc Veer, Doc Jojo, and Sir Drek. So nakakalahati na po tayo sa TAAS 3. Also greeting our partners from DOST, especially the Philippine Council for Health Research and Development, who will soon be celebrating their 39th anniversary. Congratulations, and may you have more years of funding the health research needs of the Filipino people for the next decade. So in the next decades. So also to greetings to Tuklas Lunas Fellows, the faculty and graduate students of DLSU, especially the chemistry department, friends of the CIF staff who were sort of coerced to joining the webinar. Salamat sa pagmamahal. Friends from the academe, industry, government, suppliers, thank you for attending. So lastly, to the past president of the Philippine Association of Chemistry Teachers, thank you po for gracing our event. So, okay, let's take a quick break muna. Pero while we're taking a break, anong oras na? It's time for our surprise quiz. Medyo surprise quiz with surprise prizes. So stretch na po tayo ng mga daliri natin para ready tayo mag-type, okay? So mechanics. So first one, this is open to all who registered in the webinar, okay? Make sure you're registered before you enter the contest later. Number 2, there will be 2 winners of a smart load card. Sinabi ko na pala na smart, worth 500 pesos, and 2 winners of DKSH work from home package. So you'll learn later kung ano laman nun. Number 3, we will be asking some questions. And the first one, to answer, will be asked to turn on his or her camera and mic for a brief interview. We will be monitoring both Zoom and FB Live. Okay? So lastly, we will be messaging the winners at the end of the program. So for the first question na tayo. This question is for Zoom participants. Hindi secret yung hindi na secret yung network kasi smart na nabanggit kanina. Okay? So for Zoom participants, you can win a 500 peso load card by answering this question about the DLSU Central Instrumentation Facility. So the question is, so hindi siya nakasulat sa sa slide, so better listen. In what month and year did the DLSU CIF started operating? So again, in what month and year did the DLSU CIF started operating? Okay, so pakilagay po yung answers natin in the chat box. And this is for, this question is for Zoom participants. So may sumagot na po ba? In what month and year did the CIF started operating? Anyone? So Dr. Garcia, our guest speaker, answered November 2019. I'm sorry, that's not the correct answer po. It's much earlier. And clue, it's much earlier than November 2019. April 2018, too early naman. Okay? Feb 2019, still too early. I'm sorry, this is Feb 2019, mas late na pala. September 2020, late na. October 2020, late na rin po. So it's later than April 2018 but earlier than Feb 2019. January, malapit na pero hindi din. December 2018, no. November 2019. So, okay. CIF staff, let's show the answer muna. The correct answer is October 2018. So the CIF was opened. The operation started on October 2018 when the NMR, the 600 megahertz dual NMR was commissioned to the CIF by Joel Philippines. So let's check please kung sino po yung nauna to answer October 2018. So may we request Miss or Mr. Hachi Urias to turn on his or her camera? And microphone din pala. Ayan.
I'm sorry.
Ayan. Okay, hi sir. Good morning.
Good morning.
Have you registered to the webinar?
Yes po.
Yes, okay. So, and congratulations, you won a 500 peso load card from sponsored by the DLSU Chemistry Department. Thank you so much for joining.
Thank you.
Okay? Thank you. All right. Congratulations, Mr. Urias. For the next question, we have this one. So this will be about the Balik Scientist Act or the RA 11035. So, ayun. Ito yung question natin. Please listen carefully. In what year was the RA 11035 or Balik Scientist Act institutionalized? Again, in what year was the RA 11035 or Balik Scientist Act institutionalized? Okay? This question is for our Facebook participants. So yung mga nasa Zoom po, wait tayo for the next question. Wait lang po Let us check the Facebook comments dun sa live natin. So meron na po bang sumagot? CIF staff? So again, the question is, kailan po na-institutionalize ang Balik Scientist Act? Anong taon? So may sumagot na po ba? Ano po natin? May 14 comments. So the correct answer is, so next slide please, it's 2018 po. Ayan. So 2018 po na institutionalize ang Balik Scientist Act. And the winner is Mr. Christian King Condez. Okay? So you won't be able to you won't be able to show yourself sa Zoom, pero kindly send us a message to claim your prize. So the prize is, by the way, back po tayo dun sa previous slide. Ang price po natin ay isang DKSH work from home package. So pag na-receive nyo, doon na lang malalaman kung anong laman yan. Okay? So hopefully meron siyang external monitor. Okay? And so again, congratulations Mr. Condes for winning the DKSH work from home package. Okay? So for the next question. And so another question for our Facebook Live. Again, Facebook po, Facebook Live streamers. Yan. Sabi muna natin, the prize pala again is a DKSH work from home package. So the DLSU CIF has 4 chromatography instruments, namely analytical LC, which has the UV, RID, and multi-detector. Pag sinabing multi-detector, we have the light scattering and the viscometer here. We also have the semi-preparative LC with a UV detector. We have the QTOF LCMS, and we have a Gcms with headspace. So now, the question is, anong brand ng mga instrument na to? So what is the brand of these instruments that CIF has? Okay, so this is for the Facebook participants. So let's check. Okay, so tingnan po natin yung answer. So the answer is Agilent. Okay, so Agilent po ang sagot at ang nanalo ay si Miss Rose Ann Perelonia. Okay, congratulations Miss Rose Ann. Kindly send us a message to claim your prize. Okay, so via Facebook. For our final question. Okay, so final question natin ay tungkol sa origin ng metabolomics term. So if you were attentive earlier in Dr. Lacerna's talk, you will be able to answer this question. And the prize again is a 500 peso load card. Okay? So the question is, for the fourth question, this is for the Zoom participants only. So kindly kindly make abang. Ayan. So the question is, who was the primary author of the paper of the group who coined the term metabolome, and when was the paper published? Again, who was the primary author of the paper of the group who coined the term metabolome, and when was the paper published? So dalawa po yung sagot natin dapat. Yung una yung pangalan or apelyido ng tao, ng author, at pangalawa yung taon kung kailan na-publish yung papel. Okay? So let's see what the answer is first. So the correct answer is Oliver, so yung primary author, and then this was published in 1998. Okay? So let's check the comment section kung meron pong nanalo. Okay? May we request Mr. Lauro Palanas to turn on your camera and your microphone?
Hello po.
Hi sir. Your answer is Oliver et al. 1998. Congratulations,
Yes,
you won. And pero you won pala kung nag-register ka. Have you registered?
Yes po.
Okay, all right. Congratulations. So we will be sending the details later. Okay, thank you so much for joining.
Thank you
Alright,
po.
so thank you. So we will now move on to the next part of our program. Okay, ayan. So let me introduce you to the next speaker, or our guest speaker for this event. So this will be fun, I assure you. So we are in no shortage of awesome research scientists as we welcome another researcher in the field of metabolomics. Our guest speaker is an assistant professor at the DLSU Chemistry Department and the director of the LaSalle Food and Water Institute. He earned his BS, MS, and PhD in chemistry degrees from De La Salle University, and in 2015 he was a visiting researcher at the University of California, Davis, where he did most of his experiments for his dissertation study entitled A Multi-Omics Approach to Finding Biomarkers in Philippine Civet Coffee. Okay? So for those coffee drinkers out there, this is for you. His passion and research interests are in chemistry education and bioanalytical food chemistry, publishing a couple of papers focused on coffee. Okay? So everyone, let us all welcome Dr. Emmanuel Garcia.
Good afternoon. Thank you, Chris, for that kind introduction. Good afternoon. Good morning, everyone. Di na ako nakakatulog masyado. Nagpapang-abot na yung gabi tsaka umaga at saka hapon. Can I share my screen? Okay. Here we go. So, good morning, everyone. I'm Mel Garcia. I'm a full-time faculty member of the chemistry department at DLSU. And I'm also currently heading the, as was mentioned a while ago, the LaSalle Food and Water Institute. So for this session, we're going to talk about, somehow, my experience, no, doing research on metabolomics, particularly on coffee, which you could probably think of as something that you could do for food in general. Much of the techniques are similar, as was comprehensively discussed a while ago by Dr. Lacerna. So actually, your reaction ko kanina as I was listening to Dr. Lacerna, to Karen, was dapat gawan ko na lang ng I should have just done a reaction video to her lecture, because as I was listening to her, I realized that I should have had that kind of an introduction to metabolomics before I actually did it, because during that entire lecture, I kind of, so things kind of flashed back to me, how messed up I did a lot of my experiments because of my lack of knowledge, awareness about the things that Dr. Lacerna discussed a while ago, which actually emphasizes, I hope you guys realize this, this webinar series, specifically this one on metabolomics by the DLSU Central Instrumentation Facility, would be very, very helpful for you, actually, in the sense that a lot of the things that you need to, you have to be properly oriented to this thing before you actually embark on one, which is unfortunately not the case, no, sa akin when I did something like this. And of course, unahan ko na rin kayo, it also does help that you have a facility like this one, like CIF, which is basically a one-stop shop, almost a one-stop shop center or facility for studies like this one. And that would be highlighted as we go along in my presentation. My presentation would be, I would be talking about a lot about, actually, basically about coffee, but I'll be coming off from more of a, what you call this, systems thinking approach, articulating some of my experiences along the way so that you'd be able to, pick my mind, so that if and when you have a particular interest in carrying out a study like this one, you would have some sense of reality as well with regard to studies like this one. All right. So, but I will not be exclusively talking about metabolomics, because I will be tackling, I would also be mentioning something about proteomics. So I think you already have some idea that metabolomics is one of those tools. So very, very powerful tools. So let me just rephrase that question, that title, not to omics is the solution, but what is the problem? In my particular case, of course, my study was about civet coffee, and the interest started sometime in 2007 when I encountered a coffee somewhere in Tagaytay that was rather expensive, relatively expensive, but not as expensive as I discovered it to be when I checked out the internet. Because apparently this particular coffee is quite exotic, and it sells for so much elsewhere in the world. Just like this one, this search on eBay leads us to this product, $450 for 2 kilos. So that's about 225 per kilo. Convert nyo na lang sa pesos, and that's going to be way more expensive than your usual coffee. Okay? And pardon me for using the word with an asterisk up there, but we all know that this is really literally that. Okay? And how exotic, how popular is this? In a movie, I forgot what year this was, that featured Morgan Freeman, and that starred Morgan Freeman and Jack Nicholson. The title is Bucket List. It was a kind of coffee that the character of Jack Nicholson was bragging about, because he only drinks this very expensive exotic coffee from Indonesia. It's called Kopi Luwak. In the Philippines, it's called Kape Alamid. Of course, in the English language, American language, it's called civet coffee, owing itself from its origins, which is basically a species of the animal, the civet cat. Okay? There's a lot of crap going on, actually, that it's pretty similar to. And it's nice that we're talking about this, because it's not just civet cats poop that people are drinking, but there is now a fad somewhere in Southeast Asia where the poop coming out of an elephant is also being consumed as an exotic type of coffee. Ang tawag naman dito is black ivory coffee, but I will not go into details of how different this is from your civet coffee, but just letting you know that there's a lot of crap going on as far as the coffee industry is concerned. The moment that I drank the coffee, the first time that I experienced it, while assuming that it was authentic civet coffee, was not really about how well it tasted, because as a chemist, many of us here are chemists, so our training is always about what's in it, how is it, how are we sure that this is the real thing. So immediately the question that came out of my mind back then, 2007, was how sure am I that what I'm drinking is the real stuff, the real crap. And that's one, actually, that question is basically the foundation of one of the main issues regarding civet coffee up until this day. So counterfeiting is one issue. There's because it's exotic and it is high valued, it tends to attract a lot of businessmen, and some of them are have bad intentions just for the sake of profit. So they actually just simply package their coffee as civet coffee, because no one really would notice. But again, I'm not going into details there, because even coffee connoisseurs would actually have almost an impossible. It's going to be impossible for them to be able to distinguish between a civet coffee and uh, the counterpart that didn't pass through the digestive tract of the animal. Right? And my experience, of course, was pretty concrete, because during the time when I was making my rounds in the areas where I was collecting my samples, I came across some civet coffee samples that look like this. Okay. And I don't know about you guys, but there's something that you might have noticed here. It does kind of look like something that came out of the behind of an animal, because there's clumped together. But if you found something odd here, congratulations, because that's really something that scientists were trained to do. And what's odd here is that, probably obvious to you already, they have the same diameter, and they are all rod-like in form. And you'll probably ask yourselves, is there any animal on this planet that actually poops out that consistently, that diameter, that straight. No? Parang I don't know, but if you've been observing a lot of poop, including yours, rarely does it actually resemble, parang hindi siya replicable in form, in shape. Okay? So, yan, because of that, it became more interesting for me to do this particular study, or the study that I did several years back, because there's counterfeiting. Okay? Obviously, this is not, and eventually we tested this one just for formality, but we already knew what it was. It wasn't really civet coffee. Okay. The other issues we're not going to talk about today, but I'll just mention in passing, would be disease. Obviously, because this is poop. I don't know how willing you are to drink something that's expensive, that's also going to somehow possibly endanger your life. Okay? This is how my actual samples look like, and somewhere there are rods of fake civet coffee, but and I'm showing this to you because this is also to articulate that particular aspect during Dr. Lacerna's presentation. If you have a particular problem and you're trying to answer that one, you have to make sure that your samples would enable you to answer that problem. But here, my problem is made more complicated by the fact that my samples aren't coming from the same, might not be coming from the same animal, might not be pooped out by those animals at the same time. I don't know how long those poop have been sitting on the forest or the plantation floor. So these are the kind of stuff that we don't have control over. And how do you deal with that is something that you also have to keep in mind if and when you come across these kinds of complications in your studies. Okay. Of course, another issue would be animal abuse because of the value of the coffee. Some businessmen would, in the attempt to make their products authentic, they would actually cage the animals, and this is happening even in our country. So they cage the animal, which is, by the way, a nocturnal animal, and they feed them during the day. So imagine the kind of stress that the animals are going through. No? So para kayo nag-breakfast ng madaling araw, and then nag-lunch kayo ng 6 a.m., nag-dinner kayo ng 12 a.m. Parang ganun yung buhay. No? And if you were the civet cat, you'd probably get sick. And when you're sick, you'll probably end up transferring that problem to whoever's going to consume your poop. Okay, so those are three of the most common issues that are discussed about regarding civet coffee. And it has been subject of discussion by global media that in other countries the animals are being basically abused. No? They get sick, they are force fed and fed exclusively with coffee, so it's not really good for the animal, to say the least. So that particular question, it was a very, very simple question. How sure am I that what I'm drinking is authentic civet coffee? Is this the real thing? So it was a simple question that led us to do some simple experiments at the beginning. Some of my undergraduate students picked up the idea and then did their undergraduate thesis on it and did some small specific metabolite analysis. But then eventually, as I learned more about coffee, I realized that it actually was way more complicated than I thought. That at the beginning, to be honest, I didn't think that it was something that would be befitting of a, you know, a dissertation. But eventually, it did kind of demonstrated itself to be. Okay? So the thing about this study is, of course, part of it is sampling, and the civet cat is pretty much distributed all over, you know, the country in Southeast Asia. There's civet coffee in Calabarzon. There's civet cats in Visayas and in Mindanao. And in order to carry out that study, I would have to travel. If I wanted to do something really comprehensive, I would have traveled basically around the Philippines, trying to find something on the plantation or the forest floor. And this is basically what I saw the moment I entered a coffee plantation. So, pag ginawa ninyo yung study ninyo, you submit your proposal, you're thinking, okay, sampling, I will go to a place where there's there are civet cats, and there are coffee plantations, easy peasy. Until you actually end up being at the plantation. So the moment that you enter the plantation, this is what you're going to see. Where's the civet coffee? Of course, I wouldn't want to collect samples from caged animals because I'm going to be part of the problem, and it's not going to pass the ethics review. Diba? Definitely not. So I'd have to go wild. I mean, prefer the wild than the caged ones. I have really no choice there. So this is what you're going to see. And if you're going to do a study on something similar to this, in this particular aspect, you'd better think about how to maybe do the sampling already, at least make an attempt before formulating your proposal, because you might actually end up discovering that when you're already doing it on the first step pa lang, sampling, you're going to have a hard time. And this is not an easy time. It wasn't easy peasy for me. This is where I'm going to find poop. Where is it? Did you see it? Take a closer look. And oops. And there you go. Oops. Here. Now it looks easy because I have 2 slides to show you. It's like, okay, this is the start of the search. And wow, look, there's one there. It doesn't happen that way. You actually will experience that you would be empty-handed after looking for an entire day. That's because, you know, the animals, the civet cats, don't really, you know, poop that often, and they don't poop where they poop all the time. Unlike us, not supposed to be, you go to the comfort room, so it's very easy to find where the crap is. But this one's no. So it's pretty tough. And that makes it really quite exotic. Oops, there's more here pala. Okay? So, parang gusto ko ang gawa ng ano to. I would have really wanted to do a reaction video to Dr. Lacerna's video a while ago. I mean, presentation a while ago, because every time she talks about a particular aspect in the workflow, I could actually put a comment and then share my experience on, you know, why it is important for us to understand what Dr. Lacerna is telling us. All right, so sampling. Of course, the problem here is way more complicated, if it is not already complicated as it is looking for samples. But how would you know how old these samples are? Was this poop here for what, 2 days already? Is it fresh? Was it there for a month? Was it there for a year already? We don't know, right? We wouldn't know, all right? And then on this same image, you have ripe coffee cherries on the left side on the branches. And I'm showing you that because, of course, if we're going to do a study on civet coffee, then we would also have to have some references, which means we would have to collect coffee that didn't pass through the digestive tract of the animal, which means that we'd also have to get some samples that did not go through through digestion. All right. And then you also would realize that if you're going to pick the cherries, some other questions would come out of your mind, like, if I were a civet cat, which of these cherries am I going to pick? Of course, you can think like a civet cat, but that becomes a problem as well, because there is a theory, there is a actually hearsay ito among coffee aficionados that the reason why it tastes good daw is because the civet cat picks the ripest coffee cherries. But, define ripe. And if you are able to do so, what is the definition of ripeness to a civet cat? That's another thing. Another complication would be if you don't know how to identify the types or even the varieties of the coffee, then that's going to make your study even messy. So, again, even on step 1, sampling pa lang, you really need to polish your idea, your problem, tweak your problem before you embark on this one. So yan. Okay? Anything else I want to say about this one? Wala na. So, like I mentioned a while ago, some of my undergraduate students picked up the idea because they wanted to deal with a lot of crap, or they probably loved coffee, or both. So we did some studies that basically mirrored some studies that were already existing at that time, just to verify certain theories, or hypotheses, such as differentiating in terms of surface morphology, which fortunately for us, again, to highlight the importance of having a facility like CIF, you have all of these instruments where you can bring your samples and, under one roof, basically undertake a comprehensive study like this one. You don't, of course, you don't really want to do a study where you'll do SEM in another institution and then GCMS on another. It really is very difficult, especially with, of course, this is the Philippines. Just traveling, being on the road is a problem already. Okay, so just as what an earlier researcher found out, the civet coffee has a much smoother, noticeably smoother morphology, surface morphology than, opposite. It's a much rougher morphology than than the undigested civet coffee, the undigested coffee. So that's just one of those things. And then, of course, just because we had the instrument, we did some EDX. Again, parang nagpa-plug na talaga ako sa CIF, no? Kasi when you have all of these instruments and you have the budget, and maybe you have someone as generous as Dr. Camacho, you can do a lot of stuff all under one roof. Okay. Initially, some of my students did what were doable at that level, and because the budget of an undergraduate thesis is not much, we were only confined to specific metabolites like caffeine, alpha tocopherol, right? One particular study was yung microbial naman. We did a sort of collaboration with some faculty members in the biology department, right, just to determine if civet coffee after roasting would actually have uh, uh, pathogens. No? Fortunately, for our samples, uh, the most that we had was something called too few to count. But I don't know if that is that makes you feel reassured, no, na safe yung coffee na yan. Okay? So, those few slides that I showed a while ago were the kind of things that undergraduate students did. The importance of that, the reason why I showed you those is uh, you might really want to make yourselves familiar with um, your particular samples first by doing some small experiments on it. The more frequent you study it with smaller experiments, the more comfortable you would be, and the more informed you would be in terms of handling, particularly the samples, or making sure that that particular initial stage is uh, clean, or at least with less complications before you embark on a bigger study like metabolomics. And another thing that I realized after doing these small experiments with my students was that uh, when we went out and had some conversations with coffee professionals, yung mga baristas, coffee shop owners, roasters, farmers, and you tell them what you're doing, all they would really come probably appreciate is the fact that you're a scientist. Wow, that's cool. But just like me, I ended up realizing that, wow, even if I'm doing some science, some cool science, my stuff, my results are not really that relevant to the coffee industry. Like, would they care about this? Would they care about this? Not really that much, because if you think about it, you drink coffee and you found out that it has more caffeine or less caffeine. It doesn't really matter much to you, because you know that coffee has caffeine, and if you're not comfortable with caffeine, then there's really no point in drinking coffee, right? Another realization, for example, with alpha tocopherol, you have antioxidants. You tell people in the cafes, you know what? We did an experiment and we found out that somehow the amount of uh, alpha tocopherol changes because of the fermentation process inside the gut of the animal. You'll probably be the first to realize that it's really not quite useful, because if you're after antioxidants, you're not supposed to be drinking that much coffee either, because by the time that you get the necessary amount of antioxidants from coffee by drinking that volume of coffee, you would much earlier on be suffering from the ugly aspect, the ugly side of coffee. Diba? So that's the reason why uh, at this stage, what I want to tell you is, this early, is if you're going to embark on a study that has industrial or commercial significance, you might want to, and I would actually strongly encourage you to go out there and mingle and discuss uh, heavily, deeply with people in the industry that you are going to deal with, okay? Because the stuff that they would be telling you might also be extremely valuable, just like in my case. And this is when I discovered that people drink coffee because, especially nowadays, you have really expensive coffee. Those some of you might call fancy coffee, but really the palette of coffee drinkers now has progressed, so that a bigger segment of the market now goes for flavor um, rather than just getting the buzz from caffeine. And the moment that you realize that people, more and more people drink coffee because of the exquisite, the delicate flavors, then you realize you have to study a lot more than what you used to be analyzing. Okay? And how complicated is coffee flavor? This is by no means all the reactions that lead to the production of flavors. This is basically the more significant ones, but there are hundreds, maybe thousands of flavor compounds that actually come together to give you that sensation every sip that you take from your cup. Right? So, because we realize that people drink coffee more because of the flavor, my undergraduate students back then said, okay, let's analyze the aroma compounds. So we did GCMS analyses, and boy, did we get a lot of compounds, so much that we just basically there's something wrong here, but to make it more simple lang. Because what's wrong here is that you cannot describe a flavor as being different simply because they come from different functional groups, or they carry different functional groups, or they belong to different functional groups. Now that's wrong. But for the sake of making the study a little bit more organized, but something we will delay that complicated reality later on, is by grouping those compounds, the aroma compounds, in terms of their functional class. And this is what we found. And this is basically what differentiates a civet coffee from non-civet. In this particular case, we have Excelsa and Robusta samples because this is what we were getting from the site where we collected our samples from. Okay. Now, this will also be a hard lesson for people who are working on coffee, if you haven't done much yet, because if you must know, there's several types of coffee, and especially in one type, you have quite a number of varieties pa. So that makes things theoretically more complicated, and if you want to deal with that, it's going to be very what you call this, overwhelming. All right, so yeah, so this was about aroma, but then at some point again during this journey, someone told me that this is something that's not as simple as you actually think it is. And after reading, going through some literature about coffee, around that time, metabolomics was already, quite popular, and very few people were doing some metabolomics on coffee, particularly on civet coffee. But on the year then I was about to do this one, some guy from Osaka University was already doing this for commercial civet coffee, right? But what was pretty interesting for me before embarking on the metabolomics was, this is about trying to find some markers, potential markers to authenticate civet coffee, because again, that was the original problem. How sure am I if this is the real thing? No. So one of the drawbacks of many studies is that, well, there's not many studies, actually there's less than a handful, is that the samples that were being used, some of them were really commercially obtained, no? Commercial products. And therein lies the problem, because if you're doing some differentiation, particularly authentication, you cannot run samples. I mean, it makes perfect sense naman, di ba, na you cannot run samples that you are just simply working under the assumption that they are authentic. You can't even trust people to give you samples because, like what I showed you a while ago, if I simply ask the farmer to give me some samples, ship it to Manila, I'll probably end up with the kind of stuff that are, so uniform as if it was a robot who pooped them out. So you have to really go out and take the samples yourself, just to make sure that you're as close to, if not, hindi naman as close to, you really have to get the authentic stuff, right? And it gets pretty, dirty, when you do that. But this was pretty exciting, and since it was something that was pretty cool to do, we did it. We tried to do it, and that led me to meet some guy, some awesome professor at UC Davis who was giving a lecture about glycans here in the Philippines, and I wasn't really paying much attention to glycans back then because I was already fixated with the equipment that he had that he was using. So after his talk, I told him, the guy's name, by the way, is Professor Carlito Lebrilla. He's from the chemistry department at UC Davis. I approached him and told him that your equipment are the kind of equipment that I need for my study. And fortunately, during that time, UC Davis was just months months after launching their coffee initiative. Parang half a year pa lang yata, 6 months lang. So this and this professor who wasn't really doing work on coffee at that time, for some reason was part of that initiative. I think preemptively, providentially, that was the thing for me. So I asked him if I could at least send samples, and then his reply was even better. He told me, just go here. I mean, go there. Go to my lab, which I did. To cut the long story short, I ended up there, and when I got there, they already ran their coffee science elective course, which was quite a popular, is still the most popular elective course in UC Davis. And this is the laboratory back then, a makeshift lab that was converted to basically a coffee roasting and brewing laboratory. And it was very useful for me, and this is where I got to know the top guy of their coffee initiative, Professor Bill Ristenpart, because he was at this laboratory at that time when I was looking for a place where I could roast my coffee and grind them, etc., etc. And he was pretty generous about this one. Okay. So the kind of roaster that they had was this. Again, not the kind of roasting machine that our large-scale commercial roasters would be using, but good enough for a neat science study like this one, because it is basically dedicated to roasting coffee anyway. So, of course, I need not go to the details since it was mentioned a while ago by Dr. Lacerna. We did the metabolomic analysis for coffee, for civet and non-civet coffee with these variables. Actually, let me correct myself. These are not species. These are types of coffee, Robusta and Excelsa, and the category would be in terms of civet and non-civet, and treatment would be roasting levels. Right? And for brevity, let me just show you the result of that metabolomic study. These are the compounds that showed up as having being no significant difference among the samples. Let me proceed to the next one. But when we apply one factor ANOVA, for the variables that I stated a while ago, this is what we get. For civet and non-civet, these are the compounds that basically differentiate them. Okay? So again, For the one factor, this is basically considering all of the factors but condensing them to being between civet and non-civet. What distinguishes the two would be the following: urea, civet has low, non-civet has high, and then there's lower pentitol in civet coffee, higher in non-civet, etc., etc. Pretty cool, no? But for real talk, and if you're looking ahead for the application of your study, if I'm able to identify these, and we will disseminate this eventually, even the unscrupulous businessmen would take notice, no? And the moment that they realize that they are informed that these are the chemical markers that tend to differentiate civet and non-civet, take note that these substances can be purchased. So we can, if I'm an intended businessman, I'll probably just add or extract some of those compounds just to pass what might eventually be regulation for differentiating civet and non-civet coffee. Yeah. And this is where the dilemma, an extended work came from, no? Which I'll discuss in a short while. For type of coffee between Liberica and Robusta, by the way, Excelsa is a it's a sub variety, of Liberica. These are the differentiators. So at this point, you already kind of get the idea that there are more differentiators between Liberica and Robusta, between types of coffee, than in terms of how they were actually processed, no? Or went through the animal. Okay? So you get more complications actually just by shifting from shifting what you're drinking from one type of coffee to another. And this is this is intentional. Of course, I had intentional na hindi ninyo makita, but just to show you how many of those compounds will actually be differentiators of level of roasting. And just to exaggerate it a little bit, I'm just showing you unroasted coffee, which is what we call as green coffee, versus roasted coffee. And so roasted, we have different levels pa. You have light, medium, and dark roast, and some stuff in between. But just to make it more pronounced, we compare green unroasted coffee and roasted coffee regardless of level. Okay? And this is what we get as differentiators. What all of this are telling us eventually is that we are able to complicate, you are able to get more complex complexity in coffee by roasting your coffee, okay? Followed by choosing what coffee to drink, followed by, this is far third place, having to pass it through the digestive tract of an animal. I wouldn't really tell you that this basically means that civet coffee is hyped coffee, but the science tells us that you generate more complexity by playing around with roasting rather than having it consumed by an animal and then eventually roasting it after that. All right. I'm showing you this slide because some undergraduate students from the math department naman, I should mention Dr. Rachel Arcilla, who is their thesis advisor, used the same data for their thesis entitled Discrimination of blah, 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 comparing statistical analyses. And their generalization was that based on the data that they used, PLSDA was actually the better statistical treatment out of the three that are mentioned here. Why am I showing you this? Plain and simple. Maybe it was already mentioned. It was mentioned already before, earlier today and last meeting. get a good statistician or learn statistics. Okay, so the dilemma with I can't see yung chat dito. Okay. The dilemma with the metabolomics, as I mentioned a while ago, is that it can still be tampered because once we're able to identify the differentiating compounds, the discriminant markers, is that they can be tampered because those compounds can be purchased. But what might not be, or what would be pretty difficult to tamper, would be something that we're going to find out when we do proteomics. Okay? So ito yung mga sampling ulit. So let's go to the results immediately. LCG stands for Liberica civet, unroasted. LNG stands for Liberica non-civet, unroasted. So these are the green coffee. We compare the green coffee from the unroasted coffee, the one that came from the animal and the one that didn't. And these are what we get. These are what we got. These are the peptide sequences, peptide residues, protein residues of civet coffee. And what are these residues? These are actually ones that are attributed to pathogenesis-related proteins. In other words, kind of makes sense because these are the proteins that are expressed when the beans are surrounded by pathogens. So parang yung reaction is to release proteins because of the attack of pathogens, and this is what we get. Kind of makes sense, diba? Now, if these are the kind of stuff that we're going to look for in order to authenticate coffee, then it's probably better as discriminant markers than the metabolites that we were able to identify or determine a while ago, no, in metabolomics. But then, because these are pathogenesis-related, pathogenesis response, no, proteins, the next question here would be, would there be any difference between putting the coffee beans or allowing the beans to pass through the gut of an animal versus just dumping the coffee beans in a, for example, a septic tank containing, let's say, human feces? That would be interesting. But knowing that this is a general pathogen response, might probably be very similar. And I don't know how willing people, the same coffee, civet coffee aficionados would be willing to drink coffee that were fermented, allowed to ferment in, feces of humans. But that would be fun, diba? For them. Dapat. All right. And then these ones basically are expressing us a difference between the responses of Robusta, which is a pretty hardy type of coffee compared to Liberica, but much more hardy than the very delicate Arabica coffee. You see the difference between the proteins that are expressed between Robusta and Liberica. In Liberica, they're basically none in non-civet. Okay. All right. So these are just for brevity. These are the kinds of stuff that we end up. Sorry, this is for roasted coffee, and you basically don't see much of those. Sorry, this is blow up na lang yan. This is basically what you get for roasted coffee, which is very obvious because when you roast a coffee, you basically obliterate all of the, almost all of the proteins, the amino acids na rin. Okay. So we have been doing some other omics studies already on coffee, but maybe in the future we can share with you what happens. But right now, this I think for brevity of sending the message, no, carrying out a particular specific study like in coffee, you really would have to make sure that you get a good grasp of the fundamentals, um, because especially if you're dealing with samples that are very hard to obtain, that are also quite delicate, and that are relatively unique because the way that you would be dealing with them is different. Here, we didn't do quenching because we stored coffee the way that coffee would be stored, which is basically ambient temperature, place them in ambient conditions, place them in one-way valve bags the way that coffee professionals would do them. Okay? So it's going to be a long story if I tell you how this study actually evolved, because even along the way, we had to deal with, I destroyed quite a number of columns. Mas mahal pa yung column. We had more expensive column because we're using nano LCs. And if you're not able to clean your samples...
Ito naman burger, oh. Pa, tingnan mo. Look.
Wait, what? So, if where was I? If you're where was I? Nawala ako. Oh yeah, if you're going to do to use these kinds of samples, then you would have to basically use the same knowledge and information that the industry utilizes. No? So hindi rin ganun ka textbook case lahat. No? Otherwise, you might be clean with the science, but if you're going to articulate it to people outside eventually, it becomes less valuable to them because that's not how they do it. Roasting, for example, I'll tell you that during the first experiments, mga 2007, 2008, we were roasting coffee in using a hot plate. And that will definitely not sit well with people from the coffee industry, because that's not how they do it, right? So there's a lot of messy stuff where you need to reconcile the science as well as industry practice, right? Which brings me to a question. Papatulan ko yung question na pinost do sa Facebook kanina ni Chris. Someone posted this question. Tinitingnan ko rin yung Facebook kanina. Anong sasabihin mo sa sarili mo ngayong tapos na yung experiment? All right. Which is, again, one of the more important things that I would like to highlight in this part is, of course, we already now know, probably you already have an idea how powerful metabolomics is, or omics in general is. But what really is the problem is, do we really understand the gravity of the work that we're going to do? Okay? And my problem is with this question. I don't have a problem with this question, but thank you for this question. After this experiment, you realize that the problem is beyond what you're actually doing. And we are all in this online webinar. We are in this webinar, sorry, redundant yun. Because we're all stuck at home because of this pandemic. And guess what? Where did this pandemic, when did this, or how did this pandemic start? Okay? It started from perhaps zoonotic transmission of viruses. And what makes it even problematic is that if you go around, this is a picture that I took at the Palawan airport on the way back to Manila. They were selling this product. Okay? I'm pretty sure DTI doesn't approve of this, but this was with the label of DTI. And this is medyo latest ito. Just did a search on Facebook. And because I know a lot of people in the coffee industry, it wasn't very difficult for me to find something like this. And it's still being sold. And it's being romanticized as well, because now we are even adding the dimension of, promoting civet coffee, because civet coffee is actually helping people or communities like this one. So maaantig, it tugs your heart, no? So might even convince you, the hell with pandemics, the hell with zoonotic transmission. I'm going to help a village. I'm going to continue drinking civet coffee. Right? Which is why one of the more important questions, really, when this was presented at the ASIC a couple of years ago, when I was interviewed by someone, and this, I'm showing you this article because for the teachers out there, we are doing science for the researchers and the researchers in academia. This is what matters to people. What struck them the most wasn't really the biomarker thing. What struck them was when they asked me that question: are you going to strongly advocate against civet coffee? Well, I told them I'm not quite sure, because at that particular moment, if we openly advocated, or what you call this, advocated against civet coffee and aggressively advocated against it, without properly articulating the science and the uh, connecting, no, disciplines like economics, like socio-cultural stuff, the consumers might just eventually stop, and if that happens, the farmers will, because there's no more value for civet coffee, they'll probably just kill the animals. And if you lose one species of animal, then you're messing up the entire ecosystem, right? So it's actually pretty tricky. And I'm showing you this slide just to tell you that metabolomics as a omics as a very powerful tool is going to help us, no question, finding answers to many of our problems. But we also should not lose sight. In fact, we should aggressively look beyond that problem and see how it's going to be, should be articulated to the general public. Okay? So, am I going to tell you not to drink civet coffee today? Well, not until we are able to tell every farmer that this is what we're supposed to do. Let's say we should do a study first, estimating how much we lose to civet cats, and then add the cost of that loss to whatever we end up harvesting, so that we still get the same revenue, but not actually negatively impacting the ecosystem. All right? Which brings us to the non-science part of this talk. The medyo, I hope hindi siya tearjerker sa inyo. As researchers, when you do this awesome researches, when you use these awesome tools, you have to work on the ground. And when I say work on the ground, it's not really just going there getting samples, but really picking the brain of the very people you are going to work with, and the very people who you should make that research benefit from. Right? So these are, some souvenir photos of our dealings with coffee farmers in Quezon, in Batangas. And this picture is not about farmers. These are baristas in a coffee science forum in Cebu. So we need to articulate, of course, the science, so that we enable them, we convince them, and also enable them to be part of our research. And it's a very fun experience for me because I ended up working with, I'm still starstruck today, until today, working with, national barista champions, Q graders in coffee, the kind of people would, I didn't expect I'll end up working with just doing stuff on coffee. Right? So, of course, we do not stop there. We articulate what we find out by constantly finding connections between what we're doing and what other people are doing, because that's the only way that everyone understands where everyone else is coming from. So we have some awesome stuff that would be happening, and of course I'm showing you this slide because we have a project that has recently been approved, and just to show you how important it is for us to collaborate with stakeholders of a particular industry that encompasses our research interest. Okay? Right? And I cannot say it any better, but thank you very much for inviting me to this webinar series, because I can emphasize to our teachers or researchers and industry partners that when you do a study like this one, like omics and metabolomics, you would really need a pretty awesome, almost one-stop shop um, facility. And we are all very fortunate that we have one at DLSU, manned by the best people you can get to man this equipment. Okay? So at this point, I just like to invite everyone, or well, at least inform everyone if when we are able to see face to face. I'm allow this to be a standing invitation to everyone to visit us at the LSU in our coffee laboratory. This is the small lab. It's eventually going to be big. We have really cool stuff here because we have been following what the coffee industry people have been updating themselves with, just to make sure that what we're doing inside the four walls of the university are also in consonance with what's happening outside. So pag may binila sila equipment, bili din kami, of course. Yeah. Of course, it's easier said than done. Pero yun naman yung end result na bibili. Okay? And we will be offering the first batch. We will be catering to the first batch of the Coffee Science General Science elective course, at the LSU in the next trimester. At the LSU, we will start with an online, obviously, version first. Okay? So for the researchers out there, do not limit your work to the what you have in the laboratory, but it is very important that you understand and you recognize what kind of a laboratory facility you would have. But you have to go beyond that one also, making sure that you are well immersed in the industry that houses your or that encompasses your research interest. Okay, so this picture, sorry, it looks like it's about me, but it's really about telling you that if you're doing research on coffee, you go beyond the laboratory and make sure that what your parameters are well thought of, because they are stuff that people in the coffee industry actually would agree with you. Would agree with you on, and also be able to experience the whole thing. If you have to become, if you're doing coffee, if you have to be a barista yourself, that would be better, because you would also bring with you all of the psycho emotional learnings that would keep you or fuel your interest in the study. And this photo is articulating it to the public. But in this particular photo, I have here science teachers from the basic education level. And the reason why I've always been, always wanted to have them as an audience is that I've always, thought of teachers as the perfect multipliers or ambassadors of science. No? Right? So at this point, I'd just like to acknowledge all of the people. No, obviously, top on my list is Dr. Carlito Lebrillo of the University of California Davis Chemistry Department. Bill Ristenpart, all of my laboratory mates there, the LaSalle Science Foundation, the chemistry department, everyone in the coffee industry have constantly been, up until today, sharpening our studies by making sure that our parameters are in tune with what they feel is relevant to them. And to the organizers of this webinar series, Dr. Camacho, thank you very much for inviting me here. Dr. Lacerna, for coming up with the idea of having me in the second part, which is the how and all of the difficulties and the challenges and the nuances, hidden nuances of the house. And the crew of this actual webinar series headed by Chris Argamino, and to the audience for listening to this one. All right. So at this point, I hope you did learn something beyond the metabolomics that you can bring with you as you contemplate on whatever research endeavor you have now or in the future. Thank you very much.
All right. Thank you, Sir Mel, for the kind words and the enlightening talk on the potential problems that we can face while doing omics studies. So it is our honor to have you here, and we hope to visit the coffee lab and sit in on the coffee science course soon when this is all over. So it's past 12, but we have to go on. Sabayan namin kayo mag-lunch, at kung umorder naman kayo ay sana dumating na. Ayan. So thank you for the shout out again, sir, and exposing my showbiz persona. And hopefully we can also look into the magic mirror in the future and tell ourselves that despite all the issues and challenges encountered, we are still able to pat ourselves in the back, and we are able to perform our respective researches successfully. So to recap, Dr. Garcia's talk, we were introduced to the issues on civet coffee, which include counterfeiting disease and animal disease, the process of obtaining civet coffee surface morphology via SEM and EDX. To segue, the CIF is equipped with an SEM that has a built-in EDS for elemental mapping. So send us a message if you're interested to have your samples analyzed. Coffee flavors, aroma compounds via LC alpha tocopherol content. Dr. Garcia also presented the results of his metabolomic study, which he did in UC Davis. And he was able to identify differentiating compounds between civet and non-civet coffee, including the statistical techniques which can be used to analyze the data. Coffee proteomics was also introduced, so if you're interested, we might also be able to organize a webinar on the topic soon. But for me, the most important takeaway is the realization of the importance of systems analysis. So considering the impact of our actions outside the four corners of the lab, that we also have to consider the input, needs, and perspectives of the stakeholders who can benefit from the results of our research by immersing in their communities and groups. Okay? So thank you again, Dr. Garcia, for the wonderful talk. At this point in time, we would like to begin reading and responding to some of the questions that you have posted or you will be posting. May I invite Dr. Karen and Dr. Mel to open their cameras for the Q&A portion.
Hello. Hi,
Hi, Sir
Chris.
Mel and Doc Karen. Yun.
Hello.
So, ayun po, kamusta po? Hopefully, okay pa tayo. Lunch time na, pero we have to go on with the Q&A for our audience. So, thank you again for the learnings today. For the audience, for the Q&A, we will try as much as we can to answer all the questions and address your concerns. If in case, again, we run out of time, we will respond to your queries separately. Okay, so let's see the questions from our audience and registered participants. So let's start with the questions or question from Zoom. So this is from, let me check. This is from Miss, wait lang, direct message kasi anyway. I'll check the name later. Hi, Mr. Chris. Can you ask this question to the speakers? Metabolite identification is based on available spectrum/spectra in the database of the equipment. How about those metabolites not present in the built-in database of the equipment as well as in NIST and the like databases? How are these metabolites determined or identified? So I'm posting the question for everyone's sake. And
Okay. Okay, so I guess Sir Mel, mauna muna po ako. Okay lang po.
Oo.
So in terms of yung metabolite identification, actually sa GCMS walang masyadong problema. Even though it's not built in dun sa equipment mo, actually yun nga, the databases that are available online are actually based on a specific electron ionization mode, which is ionization mode, which is electron ionization, and it's a specific collision energy, and it's very reproducible. So kahit na yung if you're not making use of the built-in database in your system, pwede mo siyang ma-identify later on using yung mga freely available databases. Medyo iba yung status when you look at LCMS kasi LCMS, different conditions, different instrumentations will give you different results. So medyo mas hindi siya comparable across different laboratories, different instrumentations. And usually, if you're really focusing on yung mga metabolite identifications like these, in some cases, you really have to build up your library. But again, this is it's a big bottleneck kasi of course these standards are not cheap, and to be able to build a library as extensive as the Metabolome will be just impossible at in the context of LCMS. And so magagamit naman natin yung mga available information that we have from the mass spec. So yung accurate mass that is obtained from QTOF, yung fragmentation patterns will also be very useful in trying to identify the identities. Although medyo magiging mas manual siya, pero these are actually very useful information. Okay, and so yun, how are these metabolites determined or identified? So in addition to these, aside from these accurate mass, structural fragments, currently there are movements into making use of the Msmx data kasi ngayon we're generating a lot of mass spec data, but if these mass spec data are actually not appearing to be significant, they are actually discarded na, kumbaga hindi na siya nagagamit, which is a waste. Kasi these are actually very useful sources of information for further expanding yung structural elucidation. So currently there's a lot of movement working on using molecular networking, especially in the context of natural products research, to be able to use yung mga available data from different sources from different parts of the world, and to make use of those information for purposes of subsequently trying to identify more of these metabolites. So definitely there's these are things that are going on right now, very exciting, and hopefully it will be of great help to us in the future.
Sir Mel, any insight on the question?
Just to add.
Yeah, sir.
Yan, kailangan seryosohin ninyo yung spectroscopy course din. No? Kasi yun nga, Even if it's not, even if it's already identified, sometimes you need to verify pa using the yung mga fragmentation patterns, etc. So it's very tedious actually na work.
All right, thank you, Sir Mel and Doc Karen. So the next question is directed to Sir Mel. So the question is, may I know about how to enroll the coffee science elective as a food science student in DLSU? Is working for the DLSU Food and Water Institute an option for practicum and as a part-time job when I take my master's degree after graduating? So this is from Jorel Alegre.
Okay, first on the first question, we are offering the elective course only to ID to 120 for the first batch. And it's not going to be a lot because since it's going to be online, we'll probably just be opening 3 sections lang, and this is for ID 120. So if you're not ID 120, that's going to be a problem for next term pa lang. But I have directives to also offer the elective course to the general public, but that would come much later kasi we wanna give first dibs to the DLSU students. Is working for the Food Institute an option for practicum? There are projects. In fact, there's one project coming up. I showed the slide a while ago. Yeah, perhaps no, but yeah, let me know when you graduate. Graduate ka muna and then let's talk.
All right, thank you, Sir Mel, for the answer. So I hope Dr. Garcia answered your question, Mr. Alegre. So we have another question. Do you issue po a certificate of attendance? So we'll be issuing one if first you have registered to the webinar, and second is that you have filled up the evaluation form, which we will be sending later yung link. Okay? So feeling ko medyo kumakain yung mga audience natin ngayon at wala na po silang mga tanong. Ayan. So
Sana all.
Okay. Sana all. Ayan. So before we end, let us invite Dr. Drexel Camacho for the closing remarks. Hi, sir.
Okay, thank you, Chris. So another session has ended, and I'm sure we learned a lot today as I do. With the second installment on Metabolomics webinar series, I hope that the knowledge and its applications you gained today help you activate your brain cells for potential research topic. You know, for our country to move forward, we need more scientists. We need more researchers. And we are looking at you right now. So by following this webinar, you have shown great interest. We want to nurture that interest in you and help you move towards the next crucial step of planning and doing the actual research. We at the DLSU CIF can help you realize your research ideas into fruition. You can partner with us, starting from the proposal stage, then we can help you on the technical aspect and celebrate with you on your publications and products. As I said before, there's a lot of research things we need to do, and we need more catching up as a nation in terms of knowledge creation and research activities. So together with DOST, let us help build a research culture in the country. And today we thank you so much, Dr. Lacerna and Dr. Garcia, for sharing your expertise. We'll continue the session on March 19, that's a Friday, for more details about how to do the data analysis on metabolomics and its application in clinical metabolomics. Thank you so much for joining us today. Keep safe always and have a nice day.
Thank you, Dr. Camacho. Thank you, Dr. Lacerna, Dr. Karen.
Thank you, Sir Mel. It was a really very good reminder for me as well. I really support that in order for us to do this good research, we do need to go out to the people who are involved in this matter. So thank you again, sir, for a very good reminder. And yun, thank you everyone for staying.
Next Saturday, tama ba? All right.
Friday, sir.
Thanks,
Okay,
Chris.
so Thank you po. Before we leave, so wag muna sana tayong umalis. We'd like to request you to turn on your cameras for a Zoom fee and to document the event. Mabilis lang po ito. Okay. So for the first page, CIF team ready na? Kindly turn on your cameras please if you allow us to I mean hindi siya mandatory but yan, if you can please turn on your cameras. So again, taking the screenshot, first one, 1, 2, 3. So next screenshot, 1, 2, and 3. Third screenshot, 1, 2, and 3. And then the last screenshot, 1, 2, and 3. All right. So thank you so much, guys, for attending the webinar, and see you on Friday, Feb 19 at 9 a.m. for the Chemometrics and Clinical Metabolomics webinar. Thank you and goodbye.
Thanks, Chris.
Salamat, Chris. Salamat, CIF team.
Thank you, Doc Karen.